So they also want to be part of this event. We're expecting them. They have gone to the airport to pick them. And I must apologize for inconveniences since yesterday that you have arrived. We actually had a change in pattern of the leadership of the university within the last 1 week. And so a lot of things changed. All the arrangements on ground changed. So I must make you to understand this. And that's why we have a lot of impromptu development. But I apologize on behalf of the management of Landmark University. And a few of them will soon be showing up, at our event, especially at the opening ceremony tomorrow. I want to believe also that a few of us here have a full travel grant. Some have a partial travel grant, but registration will be coming after this. It should precede this, but then we want to go into something just in the early hours of the day. So, and you'll be able to have access to your conference materials. But it is our culture here to begin every event with prayers. I'm sure you know that this is a faith-based university. So we just take a word of prayer from one of the participants, a volunteer. Please, yes, please. Thank you. Do we need to okay.
Let us pray in Jesus' name. Faithful Father, we thank you for this day. We give you all the glory and praise, mighty God, for this privilege to gather here. We know you're here with us. Lord, we ask that the reason we are here, we have accomplished it at the end in the mighty name of Jesus. At the end of it all, we vow to give you all the glory and praise in Jesus' mighty name. Amen.
Thank you so much. So you are the first participant to speak at this event, and we celebrate you, man. So I'm going to be introducing a few persons, especially the LOC members. Many of them are out, still running around to get one thing or the other, And then I will introduce the leadership of the Nigerian Bioinformatics and Genomics networks. A few persons that are present here. Some are present online. You can see some persons online already. Dr. Shegun Fatima is there. I'm sure everyone here may have received one email or the other from him. So let me start with him. This is Dr. Shegun Fatima. Please, let's put our hands together for him. He's the chair, of the steering committee team, and he has so much, to have this running today. So we appreciate and celebrate you, sir. And I'm the chair of the LOC team, Marion Adebi, and I have my team. Dr. Baja is not here. He's the co-chair LOC. Dr. Asani is not here. He's the chair for logistics. Okay, Dr. Asani right there. Please appreciate him. Dr. Badamosi, is he anywhere around? Okay, please celebrate him. Thank you. I have Dr. Baja just walked in, the co-chair LOC there. Thank you. I have Mr. Kweyemi Matiluko is in charge of IT, the setting up of all this laboratory, the system and all of that. He's right there in suit. Thank you. And we have members, other members. A lot of them are not here, but we appreciate everyone. I want to introduce the leadership of Nigerian Bioinformatics and Genomics Network. I have the president, of that great network here, in person of Professor Charles Adetunji, is right, right here standing, a very humble man. Please celebrate him for me. Thank you. Thank you. Thank you so much. And I have here the Vice President Logistics and Communication and, a bulk in her hands. Unfortunately, is a woman. Women can do a lot. She has delivered so much. She has been on ground since Wednesday, last week, Wednesday. So I introduced Dr. Itunu Ishewon right here. Please celebrate her for me. Thank you. Thank you so much. Then I have the representative of the Nigerian Bioinformatics and Genomics Network for South-South region. I have Dr. Lani Yotemitokwe here, right here. So he's the one representing us in the South-South region. You know, we have the 6 geopolitical zones and we have their representatives. If you participated in the election, say, hey. So only one person voted here. So we had our election sometimes ago, and these were the people that were elected. And so I have our facilitators today, Dr. Chinwe. Sorry, before I go to facilitators, one of the steering committee members is Dr. Chinwe Ekena. She's in the US. I think she's still sleeping. She's not here. So please, let's put our hands together for her. She was part of the planning all the way. And then from all the way from Uganda, I have Dr. Kweyemi Shoremekun, who is going to be facilitating here today. Dr. Kweyemi, can you raise your hand up? Okay, right here. You're welcome, sir. Also, I have Christopher Kintu. Mr. Christopher is right there. He's been going around with his nose mask all the way from Uganda, from London School of Hygiene and Tropical Medicine, Uganda. So they came from all the way. And I have Mrs. Brenda right here, also a facilitator for today. Brenda and Dawson here. They have been wonderful. They've been on ground. They will help all through today. So please ask your questions. Thank you very much. And I want to celebrate every participant here, you know, throughout the journey and all the hitches, especially the situation in the hotel. I apologize once again on behalf of the management, and I'm telling you that there's going to be an improvement today. A few things will change. I appreciate all of you. I welcome you and I celebrate you. Thank you. Dr. Charles.
Good morning, everyone. Good morning, everyone. Good morning, everyone. The house is it's too cold for my life. I will appreciate Almighty God for giving us the grace to be here this morning. And I want to sincerely welcome everyone. We have people all over the world here. We have people from Europe. Some persons came in from Russia, from Cameroon, diverse kind of, from different universities. Let me start on the existing protocol. I want to appreciate Dr. Segun Fatuma for this great initiative. Please, can we celebrate him very well? We deserve a round of applause. Because this is a very great initiative that we believe we're going to solve the global problem. Now, having said that, I want to beckon on every one of us and apologize, sincerely speaking, for all the challenges, all the hitches. We are very, very sorry. Please, I'm sure that before you leave Landmark, you are going to have a beautiful and a nice experience. Please give a smile and celebrate yourself. Please, I'm not happy with this. Let's celebrate ourselves. Don't worry with whatever that might have happened. I know, I understand. We are trying our best. And Professor Fatuma is also appealing, you will still hear from him very soon, that everything is going to be fine. So for me to be sure that everything is going to be fine, I want you to check on your neighbor. Just say, you are looking too beautiful, you are handsome. Please, let's do that. Let those things go on. Let those things go on. Let it 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 go on. Let's celebrate ourselves one more time. Let's clap, let's clap, let's clap. So I will say that I'm not going to waste time because we are behind the time schedule. I'm going to call on our amiable Vice President, Dr. Ichuno. She's going to give us a rundown of the program so that we're not going to waste time. And I want to welcome every one of you. Please network, enjoy the very full serene of Landmark University. And I'm sure the blessing of our Lord Jesus Christ and the best regard of Dr. David Oyedepo is going to follow us home. And I believe at the end of everything, we are going to be happy. Thank you very much. You are all welcome.
So I'm not going to take your time. You have the program already. I'll just say that the registration will take place during the coffee break. I noticed that, unfortunately, some people are not registered for the workshop. So we seem to have more gate crashers than others. So I'm going to be using my list to register you. It doesn't mean you cannot stay. You are very much welcome. But our packages were designed for people who duly registered. So I'm very sorry if I don't give you the bags. I'm really sorry because we have a list we are working with and we made arrangement for people who duly registered. So, but if we have excesses at the end, maybe on Tuesday or Wednesday, I'll be happy to give you the bags. But I'm going to be using my list to register all of you. So without wasting time, I would hand over to Dr. Fatima to take over. So use the program with you as a reference to guide you on all the activities we are going to be doing today. You are welcome again. So Dr. Shego, please take over.
Good morning, everybody. I hope you can hear me. Can everybody hear me? If you can hear me, I cannot hear you. Just let me know if you can hear me clearly. Okay, thank you very much. So we're going to start straight away. Sorry for starting a bit late. I will just start with the presentation. So if you are registered for the workshop, you should have received an email this morning. You should also have received the other program for today. I will just go through that quickly. Okay. So I have a slide on. So what we are doing today is a workshop that is called Genomic Analysis Workshop. And it is an introduction to genome-wide association study and polygenic risk score. So this is happening at the second conference of the Nigerian Bioinformatics and Genomic Network, which is organized by the Nigerian Bioinformatics and Genomic Network and Landmark University. This workshop is sponsored by 54Gene. So this next slide is just showing the program that you may already have. We are starting a bit late, so what I will do at the beginning is just to give an introduction to GWAS. After that, so we have a short presentation from Dr. Colum, who is going to talk to you about some career in genomics and bioinformatics for people that want to build capacity in doing analysis of large-scale genomic data. So we just hear about what are the career that are available for this kind of analysis. After that, we are going to, today you have many people with you that are going to be supporting you. You have Dr. Kweyemi Shuremekun, who's going to be taking a lead on grant there. You have Christopher Kitu, and you have Brenda Dosin. If you have any question, please feel free to ask them, or if you feel free to send an email to me. So in this morning session, we'll do some hands-on and demo. I will do GWAS analysis. We'll go for a break around 2 o'clock. So when we come back from break around 2 o'clock, we're going to do the second part of this presentation, which is going to be polygenic risk score. So this part will be led by Dr. Tinashe Chikowore. He's going to give a presentation on what is polygenic risk score and all that. And there will also be asked on. just to mention, I would like everybody to be for us to be very clear at the beginning that what we are doing today is just going to be for a day. So this course is only an introduction. So the course is not meant to give every detail that you need to be able to do genome-wide association analysis and project risk score. We are only going to be giving an introduction. So we don't expect that after today, you should be able to do all the analysis and all that. So in the future, we are planning to have a week-long course that will give detail for everything to do. So you should just assume that today's workshop is just to whet your appetite to be ready for the future course that we're going to be doing for a week. That'll be very much more detailed. So like I said, this course today will be lecture, a series of lecture, hands-on and demo, just to give you an idea of what it means to do GWAS and what it means to do presentation. So today we have some online participants. I would like to say that this course is planned to be on-site. So if you are online, we are not planning to give you any support in terms of the practical session. Of course, you are welcome to listen to the lecture, but we will not be able to support you in doing the reanalysis. So we can give you the SOP to follow if you're able to follow that, but there will not be any a lot of support for online participants. So we're going to give all the support that's available to the on-site participants. Feel free to email us if you have questions, email the instructors and also the teaching assistants. So the next slide is just to give an overview of who we are. So like I've been announced, my own name is Segun Fatuma. We have Dr. Tinashe Chikowore on online. Is here. Tinashe, do you want to put a video on so people can see your face? Okay, so we have Tinashe on the online here. We have Okwe there with you, Okwe Yemishore with you. We have two teaching assistants, Christopher Kitsu and Mrs. Brenda Udosin with you there. So today we're going to be using a lot of tools. We're going to be using the computer that was provided by Landmark University. If you have your own laptop, please feel free to use it. But please remember that we need to work on Linux. We need to have a software that is called Plink installed. We need to have a software called Precise installed. We also need to have R installed. Please feel free, if you have any question, please just let us know. So I'm just going to move now to my own presentation, which is what I find very interesting. If you have any question as I present, please feel free to interrupt me. What is most important now is that we understand the concepts that I want to present, so that when the practical session is going on, you understand why something is being done or why something needs to be done. So please pay attention to this concept that I'm presenting now so that you can understand it. So this is a genome-wide association study, and what I'll be presenting now is divided into four sessions. So the first session is going to be a general overview. Second one will be what are the resources and like workflow of doing GWAS. The third part will be on quality control for GWAS, and the last part will be association analysis. They're going to be very short session each of them. Like I said, if you have any question in the course of my presentation, feel free to just stop me and ask a question, and we just move on from there. And if also all the participants online, if you have questions, please feel free to just put in the chat or just let me know so that we can answer your question. Okay, so I'm going to start with the general overview. So in the first session, which I call general overview, the aim of this session is to explain the contribution of genetic variation in disease and phenotype. At the end of this session, I hope that you'll be able to provide an overview of the design of a genome-wide association study, GWAS. And I will also be able to narrate the emergence of GWAS. By the end, you should be able to learn what is the role between gene in disease. You should be able to learn or to be able to discuss the concept of GWAS, and you should be able to also explain the history of GWAS. So these are the intention learning outcome for this first session that I hope that you should be able to learn. Okay, so this is phenotype and gene. What relationship do gene have with phenotype, or phenotype have gene? And phenotype could be anything. Phenotype could be your height. Phenotype could be a disease, such as diabetes, chronic kidney disease. It could be COVID-19, it could be anything. So a phenotype. It could be traits like height or weight or BMI, things that we see. It could be lifestyle, like smoking or alcohol or diet. It could be other things. It could be other things like annual salary, intelligence. So these are what we call phenotype. So gene is when you are born, you have a gene that has been inherited with from your parent. And so there's always an association between your gene and a phenotype. And I've mentioned here what are different kind of phenotype that we have. So one question before I move ahead. So can you think about some phenotype of interest to you? Like, many people have different kind of interests in what they do in their research or in their life. So can anybody tell us what are the kind of phenotype that are of interest to you? Anybody want to tell us what phenotype that is of interest to you? If anybody is speaking, please give them the microphone so that we can hear them. What phenotype do you work on in your own project or your own research? Is anybody speaking?
Okay. The color of the skin.
Okay, so that's color of the skin. That's one. Any other person? Can you tell us what phenotype are you working on your own projects or your own research?
Hello, can you hear me?
Yes, we can hear you. Go ahead.
Okay, I think I'll look at HIV virus. That is a disease.
Okay, yes, HIV virus. Any other person? Another person?
Yes, sir. Height.
Height.
Yes.
Did you say height? Did you say height?
Height. Height.
Okay, I didn't hear that very well. Yeah, but that's fine. Another person?
Hypertension. Hypertension.
Hypertension. Okay, so those are different kind of phenotype, and I can see on the chart here also we can see COVID-19. Some people are more susceptible to COVID-19 than other. So the next question is that, do you think that gene play a role in your phenotype of interest? Many people, for example, they work on malaria. Do you think that gene play a role in malaria? Do you think a gene play a role in the color of your skin? Like somebody have mentioned that. Do you think a gene play a role in those phenotypes that you mentioned? Anybody want to try? Yes, somebody is raising their hand. Can you give them the microphone?
Thank you very much. I think gene, play a role in phenotype, because if you look at what phenotype is all about, phenotype is equal to gene plus environment. So gene, phenotype is a function of the gene of an animal or any living organism. Phenotype is equal to gene plus environment. So it does affect the phenotype. Thank you.
Okay. Another person, does gene play a role in your own phenotype of interest?
Gene plays a role in any phenotype, because phenotype is known as the physical appearance of a particular gene. For example, if I'm working on diabetes, there should be some genes that are responsible, either that have been deleted or otherwise. So in any phenotypical appearance, there should be particular genes that is responsible for that, either height, the color of the skin, or whatever.
Thank you. So let me ask one question. I know that many people work on malaria, and because we live in an environment that has a lot of malaria. So why do you think that some people, when they have a bite of malaria vector, and some people have malaria and some don't have, does that have to do with gene? Some, I mean, 2 people or 3 people will have the mosquito will bite them. One person will be down with malaria. The other person will not be down by malaria. What role do you think a gene play? Anybody have an idea?
Because some people may have resistance to that gene. So anytime there is infection, they may not come down with the infection.
Okay, so what role does gene play in building resistance, or is helping with protection? Anybody will give you like very specific answer to that?
For the genotypes, for some AA and AS. So the AA patient, the AA people, the shape of the red cell attracts, as it makes them prone to malaria more than AS.
Excellent. So that's exactly what I want to hear. You know, there's some gene, if we have variation in a particular gene, it can be protective for you, and it can also be harmful. Some people that have AA, they are people that have the normal normal cell. That is protective for them. But when it comes to malaria, they tend to be more susceptible to malaria than people that are AS. That's a good example. And so the next question is that, apart from genetic variation, what other factor contributes to your phenotype of interest? So we know that gene play a role, but there are other factors, and that is maybe your lifestyle or the environment. Environment play a role. For example, if you have type 2 diabetes, and because you have type 2 diabetes because your father has type 2 diabetes, you know, you can, if you are also maybe like eating bad food or junk food, you are going to be contributing more to your type 2 diabetes. So we have to understand that it is not only gene, there are other factors that play a role, such as our environment. So thank you very much for that interaction. So I'm going to go ahead. So in environment and gene, they play a role in different kind of disease. You know, there are some diseases that are called genetic disease. So what that means is that gene play a bigger role, like sickle cell, for example. Gene play is a bigger role for sickle cell, and environments play a very small role. There is something that's what we call complex disease, such as different kind of cardiovascular disease, Parkinson's disease, and all that. You will see that gene also play a role as much as environments play a role. There are others that are called environmental disease, where environmental play a role than gene. So we see both gene and environment, they play a role, but the amount of role that gene play or environments play depends on what kind of disease we are talking about. So now going to GWAS. So GWAS is an experimental design used to identify an association between a genetic variant and traits in sample from population. That is what GWAS does. We want to do an analysis on experiments where we want to find what is the association between my genetic variant and my traits, or which I also call phenotype. So I'll be using the word traits and phenotype interchangeably. So the next big advancement in the field of genetics after the Human Genome Project was GWAS. So you'll have heard about GWAS over and over again. So this is just an example of GWAS. Let's say that you are collecting sample from some individual that have a particular disease. In this case, let's say type 2 diabetes. We have some people who have type 2 diabetes. Some people who don't have type 2 diabetes. They live in the same environment. So we get their sample. We now put their sample on an array, and we do analysis to see what are the genetic variants that is contributing to them having type 2 diabetes or to them not having type 2 diabetes. So GWAS has been around for only 15 years. It's about 2005, 2007 that the first GWAS was done. So GWAS is still brand new. So why was GWAS not possible 20 years ago? Why were people not doing GWAS many years ago? There are many reasons for that, because there was no project that was called Human Genome Project, which cost 2.7 billion US dollars. That was just about 20 years ago. And to do GWAS, you first need to be able to genotype thousands or millions of polymorphism at once, which is not possible because of a gene-based microarray technology. So to do GWAS also, we need to know where are the common polymorphism, which was made possible by a project that was called AppMap project. And to do GWAS, we need a huge cohort of people, which is not possible with many projects we have, UK Biobank and all of them. So this is an example of an array. The very common one is the Illumina and the Affymetrix array. I won't go a bit more into this, maybe much later. So the first haplotype map, what they did was to get people from different countries and different continents, including 90 people from Yoruba, what is called trio, that you get the father, the mother, and the child from Ibadan. 90 people from European descent, 45 people from Chinese, and 45 people from the Japan individual, for a project that was called the HapMap project. And since then, we had the first GWAS in 2005. We have the first major GWAS in 2007, which was the Welcome to Us case control type 2 diabetes GWAS. And since then, there have been a lot of GWASs that have been done. So this slide is showing the number of GWAS that have been carried out since then, since 2005, 2007, up to date. You see that many GWAS been done on any kind of phenotype or any kind of traits. And we find so many, 250,000 genetic association from about 5,000 publications from them. So here is just showing that even though there has been so many GWAS that have been done, but they have been done in mainly European ancestry individuals. If you look at this plot here, you see, tiny here, you will see Africa, about 2% of GWAS have been done in Africa. And that is why this kind of workshop is very, very important in African countries, because there's need to do more GWAS in our own population. There's need to do GWAS in the Nigerian population, do GWAS in Cameroon, in Kenya, and all that, because these are very important for precision medicine. So a time will come that the kind of medication that will be prescribed for you will be dependent on where you come from. Are you from Yoruba or you are from Cameroon and all that? Because these are very, very important. So there has to be GWAS done in several populations. And here I'm just trying to highlight here, this is the biggest GWAS of cardiometabolic traits done in Africa. I was part of this work that was published in Cell in 2019. So we had 14,000 people that were collected from Uganda, Nigeria, Kenya, and South Africa. So this is the biggest one so far. But if you compare this to what have been done in other populations, like in the UK Biobank has sample from 500,000 people, and we have only done 14,000. So we see we have a long way to go. And there's a project in the US called All of Us Project, and they are collecting sample from 1 million people. We have only done 14,000 samples. So we have a long way to go. And there's also another project from the US called MVP, Million Veteran Program. They also have sample that is close to 1 million people. So we have done 14,000. So we have a long way to go. And that's why everybody must take this kind of workshop very serious. Learn whatever thing you can learn and look for more opportunities to learn what you can learn and begin to do our own analysis in our own country. So in that study, we'll find 10 associations. I'm not going into them. And this association that we found here, they were not found in the European population and the Asian population, because our genetic variance in our population is different from what you see in the European population and Asian population. So we must focus on also doing this kind of study in our own population. Okay, so in summary for session 1, so GWAS is about testing genetic variant across genomes of many individuals to identify genotype-phenotype association. That is what GWAS does. We want to find association between genotype and phenotype. So first GWAS was done around 2005, Since that time, there have been 250,000 association of GWAS that have been done since then. So, however, we must remember that those GWAS that have been done mainly from people of European descent. So there's need for diversity. Okay, so I'm going to share this slide and I put a link for further readings for people that are interested in that. So I'm going to go to session 2 straight away. Before going to session 2, we should take about 10 minutes. Is there any question from what I've done so far in section 1? Any question? Okay, so let's start second session. This session, I expect that at the end of this session, you should be able to recognize what are the tools that are needed for performing GWAS. This is what I want to achieve in the second session. You should be able to explain the workflow in conducting GWAS and able to understand the ethical framework or ethical guidelines that are necessary when you want to collect sample for doing your GWAS. So these are what I intend for you to understand in this second section. And here you will see, I'm highlighting here GWAS of severe COVID-19 that have been done recently, and there have been GWAS in diabetes, GWAS in anything. People are doing a lot of GWAS in different kind of trade. And this is a workflow that I want to show you. If you are performing GWAS, we should remember this. Most of the time, you start with sample collection. So let's say that your interest is type 2 diabetes. You want to know what is making people living in Omu-Aran to have type 2 diabetes, or let's say Kwara State to have type 2 diabetes. So what you will do is that you go and collect sample from, let's say, 1,000 people that have type 2 diabetes, and you can also collect another 1,000 people that don't have type 2 diabetes, and they live in a similar environment. So you collect the sample. Sample could be a blood sample or saliva or anything that can help you to be able to extract the DNA. So you process that, and you process the sample, and you genotype them on an array. It could be Illumina, it could be Affymetrix, and you do some quality control, genotyping calling, and all that. And up here, I show some software that you can use to do that kind of analysis, genotyping calling. There's one popular for Illumina called Genome Studio, and there are many ones that you can use to do genotyping calling. So this stage, many people that do analysis are not always being involved in this stage, but it's important that they should understand this stage. So I'll go back a bit to sample collection. You know, you cannot just go to people and say, give me your blood sample, I want to do analysis. We have to remember there's need for training, there's need for mobilization. So what we do in that study I show you from Uganda, you do training for staff, you go to the community where you want to collect sample, and you go to meet the stakeholders. Stakeholders could be the leaders of the community, discuss with them what we want to do. They are aware there are a lot of engagements in what you want to do. People understand what you are doing. It can include a mapping of household. It will include ethics. Ethics means that anybody that you want to collect sample from them, we get a consent from them. They sign a consent before you start asking them questions, or before you start collecting their sample. They agree that you can collect their sample. So that's why that is important. And there's so many different kinds of consent, which I will not go into them. Most popular is the broad consent, which is very, very, very popular. And there's also need to also remember that we should not be interested only in us collecting sample. We should be interested in the research participant. What we did in Uganda, we after collecting sample from them, and if you find out that people have maybe high blood pressure, for example, or they have hypertension, they have high cholesterol, so we went back to the community and support them with medication and all that. So as researcher, we are interested in our own research. We also remember that our research participants are also important. We should not just take their sample and just leave them. That would be a feedback going back to them. So these are very important, and they are also very big topic that people talk about. So we have to remember that ethics is very, very important, not only the analysis. So after doing all that, we genotype, we move to the quality control. And today, Okwe and the team on ground will be showing you how we do quality control, and they'll also be showing you how we do association analysis, which is the GWAS itself. apart from that, there are also other things that we might need to do, like genotype imputation. But this workshop will not allow us to do that today. So this is very important. And here I'm just showing you some software that can be used for genotype imputation. You know, don't forget that when you do your GWAS, you do 1,000 people cases, 1,000 people control. That is very small compared to, you might not be able to find a lot, but if you meta-analyze your study with other study, it might help you to have power to find something. So we also do meta-analysis and replication. And again, this will not be covered in today's course. And after that, we do in silico file mapping, because when you do your GWAS, what you find is an association. And we should always remember that association is different from causation. So what file mapping does is to help us find, okay, what genetic variant is causing these traits to be this, or is to be that. So file mapping is always very important. And after that, we do polygenic risk score. For example, if you know that these people, let's say, in your own study, 1,000 cases, 1,000 control, you can be able to say that in the next 10 years, these people are going to be developing type 2 diabetes. That would be a breakthrough, because if you know already from your study, we can be able to plan for those people and do an intervention for them. And that is the idea of a polygenic risk score. And that's why we're also going to cover that today, but just going to be an introduction. So can we have a subgroup of people? Can we know if they're going to have that disease in the future or not? So we should remember that, so typically GWAS is always case control for a disease, but it can also be quantitative. It can be like a GWAS for eyes or GWAS for BMI and all that. But most commonly it's always like a case control. So the individual in the study are genotyped on a commercial array, like I said, and some tool like a Genome Studio are used for genotyping analysis or calling and all that. And you now use some tools to convert them to what is called plain format that we mentioned today. So this afternoon, or after my presentation, we're going to be doing some practical session on quality control. Okay, so again, on this session, I have two guidelines of further reading on just some summary of how to do GWAS. If you want, you can read them. And also the second one on ethics of genomic study. You can read them if you want after if you have time to want to know more about tools and resources. So that's the end of the second section. Is there any question before I go briefly in the next 5-10 minutes to talk about GWAS quality control? Are there any questions? Okay, so I'm going to move. Yes.
It's not actually a question, but the slide you were presenting is as if it's not in the folder given to us.
Yeah, the slide will be shared with everybody.
Okay, thank you.
Okay, so the next one is quality.
I have a question, please, sir.
Yes, please.
Okay, thank you for the wonderful lecture. I think genome-wide association study is really interesting one, but the challenge I have using the platform is getting familiar with or having, should I say, how you get so conversant with the tools you use in doing this analysis, doing your studies. So how do you go about getting familiar? Okay, like you mentioned about, okay, maybe like arrow programming and then other platform you use in analyzing your study doing GWAS.
Okay. So one way to get familiar with them is just to keep on using them. You remember the first time you were learning how to drive and you were not familiar with where is the steering, where is the gear and all that. But when you keep on driving and driving and driving, you just automatically change your gear to gear 1 to gear 2 or gear 2 to gear 3. It's exactly the same way that we get familiar with R and Plink and all the tools that we use. For our colleagues that are going to be leading today on site, they use it almost every day and they're very familiar with it. So I would suggest that if you are actually interested in doing this, you have to use it almost every time and you just get familiar with them. Okay, so I'll just go back.
Thank you very much.
Yeah, I'll go back to session 3.
Another question.
Okay, please go ahead.
All right, thank you very much for that lecture. One of the problems we have in this part of the world is getting access to data, collecting data, and warehousing this data. Now, you've given an instance of where you went to Uganda to get data. I just want you to share what are some of the challenges that you faced and how did you surmount those challenges so that we can also use that experience here to be able to get data. Thank you.
Excellent question. Thank you very much for that question. I'd like to say that starting a study such as this, where you collect data, is not easy anywhere. It's not easy whether in Uganda or in the US or in the UK or anywhere. It's not always easy. That would be planning. Let me give you an example. For the Uganda project, with the paper I showed you that was published in Cell, from the time that the conception, as in discussing that project, up to the time that the paper was published, that took 11 years. It took 11 years. When we start saying that, can we do this study? Can we do that? Can we set up this and all that? So that took 11 years until the paper was published. So that will show you that it takes a long time if you are going to be doing good science. But I have a good news for you, particularly for people that are doing that in Nigeria. You'll have heard that this workshop was sponsored by 54Gene. And currently, our colleagues, including myself and other people in Nigeria, we are partnering with 54Gene to collect 100,000 samples for GWAS and genomic study in Nigeria. So we are collecting samples from all 6 geopolitical zones in Nigeria. So I'm telling you that as of today, we have already collected more than 70,000 samples. There's no study in Africa that have gone that far. Never, no one. But even that one has not been easy. We have a lot of people in the team. We have a consortium that is called NCDJHS. And this is costing thousands of US dollars to collect a sample. So it's not always going to be easy to collect a sample, but the first thing is that is to have some track record. And when you have track record, you're able to get to attract funding. When you attract funding, and you are able to now start collecting samples. Just to give you an example, we are genotyping some sample currently from Malawi. We are genotyping about 8,000 samples. In our group in Uganda, we are paying more than £500,000 for genotyping. You see, if you don't have that kind of funding, there's no way you can do this kind of study. So the first thing is to have the capacity to do the study. You have some track record, you can attract grants, and you can go ahead and do your study. But this kind of study they're not cheap. They require a lot of planning and funding to be able to do that. Did I answer your question? Okay, so we're going to move to session 3. And this session, the plan is just to explain a reason for performing quality control. Why do we need quality control? Discuss relevant quality control steps, and how do we recognize poor quality control in genetic association research paper. So why are we doing quality control? You remember that the capacity of GWAS is to identify true genetic association. And this depends on the overall quality of your data. If you have bad quality in your data, you will not find true association. You might find false positive, which are not what you intend to find. So we must remember that we need to do a good quality control. So the ultimate purpose is to minimize potential bias. You don't want to publish a paper and say, this gene or this genetic variant is associated with this particular disease, whereas it is false. You don't want to do that. So you must remember that quality control is key. So it also helps us to identify samples and single nucleotide polymorphism of poor quality and questionable identity. So this step, the team on ground are going to go through the step with you again and again. So in doing quality control for GWAS, this is always like the standard steps. So you do what is called sample quality control, and you do the SNP, which is genetic variant quality control. In sample quality control, you just go over the sample core rates. You find how many of those sample what is core rate for the sample. You do autosomal heterozygosity. You check for sex. You check for relatedness. You also check for confounding. In SNP quality control, you also do SNP core rates. You also do at the Weinberg equilibrium. This will be explained when in the practical session, and you'll be able to run this in your practical session. And just to give an example of what does that mean? Supposing that we have some sample 1, 2, 3, 4, 5. Let's say this person, sample 1 is Ade, sample 2 is Chinedu, sample 3 is Musa, sample 4 is Chinwere, and sample 6 is whatever person. And these are their SNPs. Maybe at this point, this person, we could not genotype this part. This person, this sample 1 has AG here. They have GG here. They have GA here. They have this one is missing here. Just an example of what we see. So you can see it's just missing. What do we do? So how do we do missing core rates here? For example, what we are doing here is missing core rates is a fraction of missing cores per SNPs. So like here, for this particular, how much of sample with missing core rate should be used, for example? If you look at this, for example, this person is missing, this sample A, for example, they're missing this SNP and this SNP. Out of 5 SNPs, they are missing 2. So we say, oh, this is like a 2/5 times 100. If you look at the sample 2, they are missing from here, that's like a 1 out of 4. This one is missing here, 1 out of 4. Another. So we have to decide what is an appropriate core rate to use for the missing core rate. But this will be explained in the practical session. And again, it seems also with how do you calculate the sample core rate? You know, the sample core rate is this way. The SNP core rate is this way. You can check how many are missing against the sample, or how many samples are missing against the SNPs. Here, I'm just showing sample core rate here is 60% because 2 are missing out of 5. Here is 80% because only 1 is missing out of 5. Here is 80%. This is 100%. There's nothing missing. Here is 80%. But this will be, you will see how this is being calculated. So again, this is a step for doing the quality control. So I'm not going to go through all this because I'm going to show that to you. How do you calculate the heterozygosity? I'm just going to go over that. Okay. You do a quality gender check. Every sample you have, supposing they have said that this person is a male, if you check their X chromosome or Y chromosome, do you also find it to be male? We have to do gender check. And if people are too related, like in Africa, for example, people tend to live together. If 2 people are too related, we have to decide whether we want to keep them in the study or want to take one of them out of the study. This also will be explained to you in the quality control. So that is just overview of quality control. I'll just go over the summary is that quality control is critical and it could be subjective the way you do it. You might say, I want to do the sample core rate first and do relatedness next. But this is a kind of very subjective. So sample QC filter should not be so stringent. For example, if you have collected sample from 1,000 people and you have some threshold that you will now need to exclude 500 people, it doesn't make sense, and that would be too expensive to exclude so many people. So SNP QC should eliminate the worst quality. When you're doing your quality control, you should remember that what you want to eliminate is bad SNPs and bad sample. In trying to do that, we should not throw away the good SNPs. So throwing the baby out with the bad water should not happen. Okay, so that is just a very short overview of quality control. Are there questions before I go to the last session? And you can go ahead with your practical session with Okwe. Any question, or I should just do the last session?
Thank you, sir. I want to ask the relationship between sample collection and quality control. How should the workflow go? Who should come first? Because the way you've explained now, it seems we do quality control after collecting samples. So if it happens that we've collected the samples and then we are having a quality control, then we realize that we've had so many bad samples and we have to discard some, then are we going to go back to take more samples? So how should we go about quality control and sample collection? That's my question.
Thank you. Good question. Now, let me use this class to explain. Supposing that there are 60 people in this class, and we have decided that we're going to use all these people, we're going to collect sample from them. That means that I'll go to each of you, I'll take your blood sample. What I'm doing at that time is I'm doing sample collection, and I'll ask you different questions. So I'm doing sample collection. So I've collected sample from 60 people. I've not done any quality control. So when I get your sample, your blood sample, so I go to the lab, I do DNA extraction, and I also do genotyping. Pay money, do genotyping, I have the sample. So now I have the sample. Now I want to do quality control. So I check, I say, okay, there are two SNPs that this is the SNP core rate for Ade is 70%. This does not meet my threshold. So I'll take Ade out. So now I have 59 people. So I will say, oh, Chinyere, okay, it's fine. Oh, Musa, okay, it's fine. Chinedu, it's not fine. So out of the 60, maybe eventually I will say I'm going ahead with 55 people because I have good quality. Is that clear? So I will do the quality control is what would that decide? Am I going to go ahead with analysis of those 60 people I've collected some from them, or I will need to exclude some people that maybe their sample is not good due to the way it was collected, or it could be due to any other reason. So that's the way I will answer that question. Any other question on quality control before I go to the last session?
Thank you very much for this lecture, sir. I really want to understand the meaning of what you mean by read and then cause in research. Maybe you've done a particular sampling and then you want to, you will tell you, okay, quality control or quality read and then all of it. What does it really mean, sir?
Quality read, you say read?
Yes, read and then calling. What does it really mean? When you say, okay, you're doing calling, and what are you really doing?
Okay, so I think what we are explaining here today is a genome-wide assertion study. And when we do genome-wide assertion study, mainly we do with genotype, which is different from when you do whole genome sequencing, where you start doing read and all that. Whole genome sequencing is like you want to find what is the ACTG block for every location in an individual. But when you're doing genotyping, you don't find that ACTG for every location. You're only looking for interesting places. Let me give an example. You know, the sequence, the whole sequence of each one person is about 3 billion. But when you're doing genotyping, you're not trying to type all those 3 billion. You're only interested in where there are variation. Let's say 1 million or 2 million, 2 million. So you are targeting those 2 million. And that's the reason why at some point there might be need for you to do genome imputation, because you're not targeting every location. You are only interested in variation, because that is what is important for GWAS. I think your question relates to when we do whole genome sequencing, which is not what we are doing today. Any other question on QC?
Yes, thank you very much for that very good lecture. Personally, before coming to this workshop, I'm not into genomics or bioinformatics, but my thought was that whatever I take out from this workshop would determine whether I should go into bioinformatics or not. And so far, I think you are convincing me to take that step. The question I want to ask is this. When collecting these samples, from the sample you gave us, is there no need to also collect medical history or medical condition of for each sample? I am looking at the possibility of applying machine learning to this data to do some prediction or to do some classification. So if you don't collect medical history, do you think collecting medical history would be a good thing? Looking at the applicability of machine learning to the data for classification. Thank you very much.
Excellent question. That's a very good question. You see, the more information you can get, the better. And a good standard for GWAS is UK Biobank. In the UK, they have what is called UK Biobank. And that UK Biobank is linked to the electronic record of each person. They have sample for 500,000 people from the UK. And it's linked to, you know their height, the color of their eyes, apart from knowing whether they have sickness or whether they have cancer or whether they have disease or whether they have anything, you also have information about a lot of things about them, which is very important. You can find out, okay, do people that have black color, are they the people that are more likely going to have this kind of skin cancer or something like that? You can find a lot of information. So I agree with you that there's need for us to also collect other information, not only what you are interested in. In that study that I show you from Uganda, we collected so many information. We asked people about cholesterol. We asked them about lipid traits. We asked them about food blood counts. We asked them about their height. We asked them whether they smoke. We asked them about their diets. We asked them how many fruits they eat per day. We asked them all kinds of questions. The WHO has a standard questionnaire that you can use for this kind of study where you can ask many questions. So this is very important. And if there's electronic record, it's even much more important. Okay, so any other question? Yes.
Thank you very much, Dr. Fatima, for that brilliant lecture. I want to find out, when you talked about data collection, even in Uganda, and you talked about how much grants you got and how it's tasking, it took 11 years to even publish from that exercise, and then the funding that is required to even collect data ordinarily. So are there existing data banks that we can easily get data for GWAS analysis? Because when I saw some of the abstracts that were sent to this workshop, I found out that most of them were wet lab stuff, and we're like, those of us in dry lab, so to say, computer science, how do we then come in? Because I'm so enthusiastic about this workshop that thereafter, with all those genomic analysis tools and ideas that we bring from here, we should be able to publish thereafter. So are there existing data databases on GWAS that we can walk into and then carry out some analysis and be able to publish thereafter? Thank you.
That's a very, very, very good question. Because if you want to do GWAS and want to show like a funder that you can do something, you have to do it first before the funder can say you have a track record. So the best way to start is to work on existing data. You build your capacity, you can do analysis, and you look for existing data, you analyze them, you publish. So you are showing people that I can do this thing. So when you show that kind of skill that you can do it, you now go to the funder, you say, welcome, see this paper that I published, I can do this analysis, I need to give me £200,000 to collect sample. That is the best way to do it. And there are data that you can use. I mentioned UK Biobank. UK Biobank has 500,000 people from the UK. And this data can be made available for you for free. Or you pay like maybe £2,000 or something that's not very expensive. Some people pay, or you collaborate with some people that already paid. Our group, for example, we have access to UK Biobank. We have access to MVP, about 700,000 people. We have access to many data that we work on. So I think maybe one way to start is to collaborate with people that have access to this data set. And you work with them, you publish, and when you publish like that, you are showing that I have a track record to do this thing. And you can attract funding. Because to get funding, you need to show track record. So thank you very much for that question. I think I made a mistake at the beginning. I didn't ask people to say their names when they ask questions. So I couldn't see everybody clearly, but I think that you guys are asking very, very good questions. Any other question before I move to the last session so that I can allow the team to go ahead? Okay, so let's.
Sorry.
Okay.
One last question. In the previous slide, you shared for further readings. I just want to call your attention to the fact that maybe you could just copy and paste some of those texts in the chat on Zoom such that we can have access to them, and maybe so that we can have it on our notepad.
Yeah, we share this slide with you, so you don't have only the further reading, you have everything.
Okay.
So the team there will share the slide with you to use.
All right. Thank you, Mr.
Great. Thank you. Okay, so the last session is association analysis, which is GWAS itself. And so at the end of the session, you should be able to describe some statistical method for identifying phenotype, genotype association, identify what kind of model is appropriate, and what do I need to adjust for, and how should I interpret whatever plot that comes from the association analysis. And again, just to mention that in GWAS, the whole purpose is that we are finding association between your gene or genetic variants and phenotype. And I give an example here. If you have a SNP that is called RS334, is this phenotype, this variant, for example, is it associated with type 2 diabetes? So we identify SNPs where one allele is typically more common. If you have cases, people that have type 2 diabetes and people that don't have, maybe in your analysis, you find that this particular SNP, for example, is common in people with type 2 diabetes, but it's not common in people without. That should give an idea that, okay, I begin to see this genetic variant a lot in people that have this disease, but it's not here. That's an example of showing you that there's an association between that SNPs. So if you are doing a case control, so case control is that you have people that have diabetes, people that don't have diabetes. So you use logistic regression. That's statistical method will be logistic regression. If you are working on continuous traits or continuous phenotype, an example is if you go to the hospital and you say you want to do HbA1c, HbA1c is also testing for type 2 diabetes. What they will tell you is that your HbA1c is 4.5 or 6.1 or 6. You don't classify people by you have diabetes or not. Just put their value there. Or if you are doing GWAS on height, for example, so you think about a linear regression because it's a continuous trait. And here again, to show an example of HbA1c and these particular SNPs, find the association. We must also remember other things such as ancestry or people. Most of you that are into biomedical science, we know more than me that if you have sickle cell traits, if your genotype is AS or AC, for example, that might affect your HbA1c. You might go to the hospital and say, and they might tell you that you have type 2 diabetes, whereas you don't have. And the reason is because they are using a tool called HbA1c to do the diagnosis. And that might not be true. So you have to do fasting glucose for you to be able to know your true state of type 2 diabetes. So we must remember that there are sometimes you need to adjust for some covariants, such as age or sex or ancestry and all that. One model that we use a lot is a linear mix model. It's very powerful to adjust for ancestry and relatedness in individuals. Okay, so in GWAS, all we are doing is that I want to assign p-value to every variant. And because we have so many SNPs, and we have to also remember multi-testing. So we always use a p-value of 5 times 10⁻⁸ to define an association between a genetic variant and our traits. And here I try to explain how we got that value. Normally p-value is always 0.05, but because we have like maybe 1 million independent SNPs, so we have to use this stringent significant level. So the tool you'll be using today is Blink. I'm not sure you're going to use GEMA. You're not going to use this, but these are some of the tools that are used for performing your association analysis. And so when you do your GWAS, what you want to see is a Manhattan plot. So Manhattan means it just in New York, you see all this very tall story that is coming from there. Very tall plots showing like here you can see the S-axis here is showing the chromosome 1 to up to 22, and the Y-axis is showing the minus log or the p-value. So if you see all this one here, they're showing the SNPs, the dot are the SNPs, and all this one are showing that this one are kind of associated. A line here is showing the line of association, and all that. So the team on the ground will explain more to you about the matter plot. And also, sometimes you can plot the Zoom plot just to see a portion of your association. This is a QC plot just to show the quality of your data. Okay, so in conclusion, so when we design GWAS, selection of the appropriate association test depend on the number of factors. What kind of association test are you going to be using? That depends on the type of phenotype. Is it case control or continuous? It depends on what you have to control for variants. For example, am I controlling for population structure and all that? And if you want to visualize them, you can use Manhattan plot, you can use Sticky plot, and also regional plots. Again, I have a further reading for people that are interested, and that will be the end of my own presentation today. I'm very happy to take any more questions.
Yeah, thank you very much for that. Another beautiful session from you. My name is Amos Badger.
Okay.
I want to ask, what are some of the research issues that I have in terms of determining this association between the genes and the diseases? And then also, I want to ask is this issue of correlation, because the two algorithms I heard you mention, logistic regression and linear regression, the underpinning concept is correlation. And we have this issue that correlation doesn't really mean causation. Two things correlate doesn't mean one is causing the other. It could be some other confounding effect underneath that is causing that. Do you have this as a research issue in association? And what are some other research issues that you have in this area? Thank you.
Great, very good. I mentioned that a bit earlier. So when you do GWAS, what we are finding actually is association, which is what you call correlation. We are finding association because a GWAS does not help you find causation. And if you look at the workflow that I show, there is a tool that is called fine mapping. So if you look at that, the fine mapping, what fine mapping does is that if you are saying that I find these 10 SNPs at this location, they are associated. So what fine mapping does is to help you find which of them is causing the disease. So we have another one, which we're not going to cover today, is fine mapping, that is helping us to find the association. Okay. So any other question before we move to the next session, just to listen to the career advice on what to do with all these jewels. I'm going to play a video by...
Excuse me.
Yes.
Sorry, I'm Tinuke Oladele from Unilorin. Sorry, we want to have the soft copy of your lecture.
Oh, madam, okay. Yeah, it is you. So thank you very much for joining the class. I'm going to share the lecture, as I said earlier. I'm going to share with you. Okay, so I'm going to play this video from our colleague in the US, trying to advise us, and also part of the 54 Gene team on what to do with what we are learning today. Let me know if you are hearing him clearly.
I'm Ogdulana. I'm head of the Genomics and Data Science group at 54 Gene. I'd like to present a few slides to you today about a career and opportunity in genomics and bioinformatics, and just give you a sense of my journey through this field over the last 20 or so years. Okay, and so I have a few talking points, just 4 or 5 slides. Why did I choose bioinformatics and genomics as a career? Why are bioinformatics and genomics, in my view, great fields to become an expert in? A personal history of some of the GWAS efforts I've been involved in, and then I'll end with what I view as some of the benefits of genomics, namely public code and data sets, space for creativity, is a vibrant research community. There's a lot of diversity and ways to do things, and so I'll touch upon some of these things. And so, yeah, why did I choose bioinformatics and genomics? So I was finishing a degree in biotechnology, and I had some wet lab experience, and I preferred, excuse me, I preferred the idea of a dry lab. I preferred the idea of iterating over data, exploring data, mining data, rather than kind of the formality of the wet lab. I wasn't particularly good in the wet lab either, so that helped. And then, I like the idea of computers and coffee, and so it seemed like a very natural fit to me. And bioinformatics back then was kind of different. There was a lot of alignment, multiple sequence alignment approaches, a lot of exploring the genomic data as it began to emerge for the first time, as the Human Genome Project was still, ongoing at the time. So it was a very different time, but it was a very, very interesting and exciting. Why do I think bioinformatics and genomics are great fields to become an expert in? There are a lot of reasons beyond what I list here, but there are lots of free tools. You don't have to get necessarily a license for many of these tools. There's a lot of free data out there. You don't need to generate your own data. There's a public data, public results, public GWAS results, for example, from the UK Biobank. It's also, in terms of the research being conducted, it's moving towards very open access. bioRxiv is leading the way there. And you really, anybody can get access to the latest and greatest papers as they are being submitted. And so that's there's great equality there, and anyone with motivation and can really, keep up with the field. There are great community networks and forums like Stack Overflow is one where you can debug a problem. There's really solutions to nearly every problem out there, and you can really, much of the genomics can be self-taught. Not all, but you can really learn a lot just looking at these resources. There's also multiple disciplines in one, right? You learn biology, genetics, data science, statistics, machine learning. And you can reuse those skills, and even if your career evolves into different areas. And yeah, you don't need to always be physically, in a lab. It is kind of a virtual science of sorts. So a personal history of GWAS. When I started, it was at the end of kind of a candidate gene era of target discovery, and so for schizophrenia research, which I was doing at the time, a lot of people had their favorite gene and a small sample size, and they were just testing for an association. And so there wasn't a kind of an unbiased assessment of an unbiased search for genes that caused risk for schizophrenia. And so when genomic arrays and kind of the real era of GWAS kicked off, it moved away from like picking a gene and having a kind of hypothesis to more of an unbiased, hypothesis-free data exploration exercise. And then with that came thoughts about how could we adjust for things that confounded our observations? How could we, what are important in biasing the outcome? Age, sex, principal components for ancestry. Things like that. If all your cases are from one country and all your controls are from another, that's a problem. And so figuring out all those things were going on. And then PRS came along, and PRS came from animal livestock genetics. And the idea there was, how can you improve livestock using these risk scores for the quality of the breed. And in the case of GWAS, the idea was, what is the global burden of risk alleles in people with a phenotype versus not? And so I was involved in some of those over a decade ago, and it kind of went quiet for a while, and now we can all see it's a huge part of everything that's being done in GWAS, many things. And so, yeah, just to reiterate, it went from candidate genes to kind of early microarrays with 100,000 or a few 100,000 markers along the genome, to then much more granular whole genome sequencing that allows us not only to look for common variants that might tag a gene or kind of point you in the right direction of where a disease variant might exist, but something much more granular like whole genome sequencing that allows you to actually type the deleterious variants. And so, yeah, and similarly, that's on the kind of technology front, but then versus now in terms of tools, data, computational environments, all that has changed incredibly rapidly. Tools are much faster, multi-threaded. There's we have cloud-based environments, and the data can be stored, compressed, accessed in much more rapidly and efficiently. So it's been remarkable to kind of see that change, and I think it's much easier to do things efficiently now, and there's just so many resources out there that are cheap, if not free. So that's great. Okay, so yeah, and then lastly, I'll just end with what are the benefits in my view of genomics. There's so much publicly available code and data sets out there. There's space for creativity. There's a vibrant research community. There are many methods and tools. And the data set can be analyzed in many different ways. You can ask a question about selection, or you could ask a question about admixture. And if you phenotype data, you could run a GWAS. And so, the sky's the limit really, and you can really make great use and reuse of all the tools and data that you have at your fingertips. So I'll leave it at that. But I wish you all the very best of luck. It's a great choice. It's a great field. And yeah, please do reach out if anyone has any questions. Thank you. Bye-bye.
Okay, so that was a presentation by Colum. Colum is the Vice President Data Science for 54Gene, and is trying to advise on anyone that is interested in doing genetic analysis or doing bioinformatics kind of career paths. If you have questions, let me know. I can try to answer them, or I'll pass them to Colum to answer them. So after now, what I think we'll do is we can go outside where there is a banner and take a good photograph for this morning, just now. After the photograph, we go for coffee, and people that have not registered should do their registration. And we'll come back for the practical session that will be led by my colleague, Dr. Okoyemi Shuremekun, and others over there. That is what I think we should do now. Is there any announcement that I missed? Do we have Dr. Shewon there, or any of the LOC there to give us any further announcement, or that can lead? Okay, we have Dr. Charles. Can you help us coordinate this? Can we go out now for group photograph, coffee, and registration? Dr. Charles, can you hear me? If you are speaking, I can't hear you. You have to use the mic.
Okay, what's the next thing now? Photograph.
So next thing is group photograph aside,
okay.
Coffee and registration for those who have not registered before. I will come
that's
back
very
to the
good.
practical session.
Yes, coffee is ready now. Probably maybe we'll start with a group photograph. After that, coffee, then we go ahead with the registration.
Exactly. So let's do a full photograph first before we do coffee and all that thing. So
all right, thank you very much.
Yeah, thank you very much, everybody. The team over there will continue with the practical session. Bye-bye.
All right, thank you, sir. I think you can go ahead with the group photograph now. Mr. Jimmy, how are we going to do it? Is it outside or
the online session, online group? Please, can you also take a break?
Please, let's move down to downstairs. Sir? Entrance. Okay, so please, let's move down to the entrance. Where do you have the banner so that we can take the pictures? That please. So after that, then we move to the coffee. We are coming back. The coffee is outside. So please, let's move down. Let's move down. Mr. Jimmy, where are you? Where is Aran? Let's take the group photograph first. Let's move down. Let's do that snappily. The coffee is ready so that. So you may leave your bags and property, everything is saved. Your bag, no problem. No problem. Nobody will assign. Please, let's move down. Let's move down. There's no time.
Hello, everyone. We're going to be starting in the next 3 minutes. Please, those outside, please, can you come inside? As we'll be starting very soon. Hello. Hello. Okay, yeah. Yeah, good afternoon, everyone. Yeah, we're going to be starting the workshop now. Please, those still outside, may you please come inside as we're going to be starting now. Good afternoon once more, everyone. So this afternoon, we'll be doing the quality control workshop. Quality control. And then afterwards, we'll be doing association studies. So during this session, my name is Opeyemi Shuremekun, and I'll be assisted by Christopher Kintu and Brenda Udosen. All right. So I'm just gonna give. So I'm going to give a little bit recap. Yeah, okay. So I'm going to give a little. Okay. Yeah. So I'm going to give a little recap of what Dr. Fatimo has shared about GWAS data generation. And then I'm going to move to the types of ping file. And then we'll go on to the hands-on proper. So this slide here is just showing how you obtain your data. For example, you're trying to do a case control study for individuals that have perhaps a particular disease of interest. Maybe this individual have cancer or diabetes. Maybe they have cancer or diabetes. So you extract their blood, then afterwards, you extract the DNA in the blood sample. Then after you must have extracted the DNA, you prep them on an array, which you then take home to your genotyping platform, maybe Illumina or whichever one. Then after which you have genotyped the data, then you can then go on to quality control and then mine the data and then tease out some interesting findings from your data. All right, basically, because we're using Plink for this quality control, I'm just going to go through the major Plink formats that you'll be encountering during this workshop. So we have three basic Plink format file. We have the farm file, BIM file, and bed file. So the farm file is just a file that contains information about the individual, while the BIM file is a file that contains information about the genotype. So I'm going to be showing you how it looks. All right. So while the bed file also contains information about the individual and also genotype information. And then while farm file and BIM file are readable, like you could just open it up and then you see the content of farm and BIM file. Bed file is not human readable. So you just see that you try opening it and you just see some things that you could not read with human eyes, but it's actually readable using your computer. Yeah, so this is how a farm file looks like. Like I said, farm file is a data that contains information about the individual in that particular study. So most farm file always contain 6 columns. All right. So the first column is a family ID. When you're doing a case control or a population or a family-based study, so you have a family ID. And the next column is the individual ID, which is very important compared to family ID because you can actually get a lot of information from the individual. So the next column is the paternity ID. Yeah, so the paternity ID. Then after that, you have the maternal ID. Then you have the sex of the individual. So the way it works is one is coded for male. So if the individual is a male, you have 1, but if the individual is a female, you have 2. All right. So the last column is the phenotype. And then should in case the sex of that individual is not known, Plink will record it as 0. Then the next one is the BIM file. Like I said, the BIM file contains information about the SNP, that is the variant. So the first column is the chromosome. You have 1 to 22, then some will have the X and Y chromosome included. Then you have the SNP ID. Sometimes you will see RSID, while sometimes you will see the chromosome number joined together with the base pair position. Then you have the genetic distance, which basically measures the divergence between ancestry, between different populations, and sometimes between species in a particular population. Then the next column is the base pair position, which tells you the actual position of that particular SNP within the DNA. Then you have the allele, the minor allele and the major allele, as the case may be. So like I said, bed file is not readable, so you won't be able to see the bed file here. Yeah, so just want to show you the importance of doing quality control in your data. The first image you're seeing here is both of them are Manhattan plots. After you must have done your association analysis, You see this Manhattan plot just for you to have a picture of what is going on in your analysis. So the first one you're seeing here is a data that was not quality controlled. So you will notice that there are quite a lot of noise inside in this data. If you look at that significance line, and then above it, you're seeing some variants, mobile hop. Just so you may know, those dots you're seeing, each of the dots represent a SNP. All right, and then what you have down there are the chromosome position for that particular SNP. While on the other side, what you're seeing there is the the negative log P, which is a function of the association of the genetic association of that particular SNP. Now, down is a data that was quality controlled. Both this image were taken from the same data, but the first image was a data that wasn't quality controlled, while the second one is a data that was quality controlled. So you would see that it's quite very important for you to do quality control for your data. And there are different ways in which errors might come in your data. For example, you could have batch error. Maybe you're trying to genotype your data in batches, right? And then at such instances, there might be some conditions that vary amongst the batches that you have. So that is why it's very important for you to do quality control. Another major reason is that during data handling, you may just mix up the data. Maybe an individual A is a female, then you mistakenly label that individual to be a female or to be a male, whichever case may be. So you see, it's very important for you to do quality control, so you can be sure that the association that you're getting, that they are true causality. You're not getting false positive. All right. So whatever you're getting after your quality control, you're sure that, oh, the association I'm getting or this particular SNP is truly associated with my phenotype of interest. So that is why you do quality control. So that's just a recap of what Dr. Fatima has done concerning his lecture on quality control. So now we're going to be going to the practical, the practical now. So earlier we shared a particular folder. All right. So in that folder, the folder is called GWAS. I believe most people have that folder. All right. So in that folder, you have most people have this folder. So you see inside this folder, we have a simulated data that we're going to be using for this practical. And it is named AppMap R3. So you will see that you have the BIM file. Okay, so you see you have the bed file, the BIM file, and the farm file for that particular data set. So that's what we're going to be using. And we've also included some R scripts, so you don't have to stress yourself running those R scripts. So what you just need to do is just to copy and paste some commands, and then we're going to be explaining what those commands are doing, and then we'll look at the result, and then we'll pick it up from there. Now, for this quality control, we'll be using Plink. Included in that folder that has been shared with everybody, we have Plink put inside that folder. So all you just need to do is to run Plink from that same folder. For people who have Plink installed previously before this, and you probably installed it on your root directory, so you might actually have to call it up from there, then put the path to Plink. But for people who have this data, this folder, you have everything installed in this folder. And then also included in this folder is the documents, which contains all the scripts that you're going to be running and what the scripts are doing. So I'm just going to show you that now. So I believe everyone can open this file and you see this file. So the commands we're going to be running are colored in red. The font color is red. So why those ones in blue are just comments to tell you what those commands line are doing. Okay. So I believe everybody has these, and then I'm going to be starting the demonstration now. I have, like I said, Chris and Brenda are going to be assisting you to walk through it. So I'm going to share my screen and then show you the demo. Should in case you have a problem viewing it, so we have people who can help you, and you can actually signify as well. So on your computer, if you can launch your terminal, if you launch your terminal and then you move to the folder that we have shared earlier, the GWAS data, the GWAS folder. Hello, everyone. So on your machine, you will see a terminal at the bottom right corner. You can launch your terminal from the bottom right corner. So the GWAS folder that we shared earlier, for easy accessibility, it's better you just move it to your desktop. If it's in download, or if you can work from your download, it's fine. But for easy accessibility, the GWAS folder you downloaded is probably in your downloads section of your computer. So just move it to your desktop, like just drag it to your desktop. All right. So once you drag it to your desktop, then you can then launch your terminal and then move into that folder. When you're in your desktop, just do CD. So for those that are probably not conversant with the command line to use, so for you to move into that folder on your terminal, just use CD space GWAS. So the name of the folder is GWAS. So just use CD space GWAS. So it will navigate you into the folder. CD space GWAS. For the CD, you're using small letters, not capital. CD, small letter, then space GWAS. The GWAS is capital letter. So I believe majority of us have moved into the data. So I think we can move to the next step. I think 80% of us have everything set up now. So we will move to the first quality control. So the first quality control we're going to be doing is missingness. Like what has been explained, we have two types of missingness. We have per sample missingness and per SNP missingness. So I'm going to be sharing my screen and I will show you that. So what per sample missingness mean is sometimes when you do your genotyping and you get your data, you'll notice that in your data there are some individuals that are missing. And then when you try to do analysis with such individual, you might be getting false negative results. So it's good practice for you to always delete such individuals that are missing in your data. That is what we call per sample missingness. All right. So the other one is per SNP missingness. Sometimes when you run your doing genotyping, there are some SNPs that will be missing in your data. And this could actually be as a result of genotyping error. Sometimes the genotyping platform that you're using is unable to call that particular SNP. So because it was unable to call that particular SNP, it's going to put it like 0, which means that particular SNP is missing. So it's like I said, it's always good practice for you to delete such SNP. So the thing we're going to be doing now is I'm going to be running the first quality control, which is per sample QC and per SNP QC. So you're going to see it on the projector now. So like I said, we also shared a document that has all the code that you'll be running. So you just need to copy that code. And while you're running that code, I'm going to explain what each code is doing. All right. All right. So I'm going to run that now. So on that document, you will see where we write 1, determine SNPs and individual missingness. All right. So just copy the red command starting with blink, and then just paste it there and then run. All right, you see what's happening on my screen. I'm going to explain what that is. So the first command you're seeing there is blink, which basically means that you're trying to call the program called blink. You can see the highlighted one, that is blink. Then the next one is a flag called bfile. So this file is used to designate your data. In this case, our data is named appmap 3 underscore R3 underscore 1. So that is our data. So you'd see we use the B file to call that data. So perhaps you're working with a different data. You could actually change the name of the data, but what remains constant is the B file and the name of your file. All right. So after that, the flag in blink that is used to determine missingness is -- missing. So once you put -- missing and then you press enter, you'll see that your data is going to run, and then it's going to generate two main outputs. All right. Like I said, you're doing per sample QC and you're doing per SNP QC. So it's going to generate two data, and then let's see those data. Have you run that command line? So we are having two outputs, right? So the first output is blink lmiss, which basically means blink locus missing. It designates this missing SNP in your data. So I'm just going to open this data, and I will see what it looks like. Okay. So when you run that, for you to view that data, all right, just use add blink.lmiss. So that's going to show you the first 10 line of what is inside that data. So when I do add blink.lmiss, you see what we are having there. For people who that blink did not run, just put /blink before you run blink in your, then it's going to run. / before that command line, /blink. Yeah. Yeah, now copy the. So, like I said, for those that have that blink not running, just use // then copy the command, and then it's going to run blink. So just run the program. So we copy this. You have to copy this.
You just show me.
Copy that. No, you have to copy everything. Copy everything. Copy everything. There's a document that was shared. Open the document. So just copy this. But before you copy, put dot forward slash and copy the answer.
Dot forward slash.
Yeah, dot forward slash. Before please. So you didn't put up, you have to put a flag. So put that forward slash, dot slash, and copy. Everything before you run. No, don't say it yourself. I'm coming. Sorry, I'm coming.
Talk about getting the head.
Yeah, so you have the data generated. Yeah, good. Can you do LS? Yeah. LS minus, no. Yeah, it's fine. Can do that. You just do H, think L. Okay. And get it. No. Check it on for fine.
This also.
Yeah.
I'll carry this one to
So Tiron, can you open your family? Okay, so Emma, you really cool. You are very sensitive. So that's one of them. Just put no, it might carry on down after now. What is the man? Windows. Do you have the data here?
Yeah.
So where is the data? Can you just minimize this? All right, I believe we all have the file generated. We've all successfully run the first command. All right, if you do ls space minus lrt, like I said, it's going to generate two files. So you see the last two files that your command generated. Yeah. So what you're basically looking out for is .lmis. So if you do add, so you can just type add. So I'm going to show you. So if you check, if you do ls space minus lrt, so this is just for you to list your files, the files on your in a particular folder based on how they were generated. Okay, so when you do ls minus lrt, then enter, you will see that the last three files are plink.lmis, plink.imis, and then plink.log. So the plink.log just shows you information about the command that you ran and the files that were generated. So what we are basically interested in is the plink.lmis and plink.imis. Don't forget, the main reason why we're running this command is for us to calculate the missingness in our data. So we are trying to calculate sample missingness and SNP missingness. All right. So what you need to do now is let's look what is inside this file. So when you do add, all right. So do add space add space plink.lmis. Yeah, just copy the last file, plink.lmis, when you did ls minus lrt. So you should see the file there. All right, everyone. So if you see what's on the screen, when you did plink.lmis, like I said, what you're trying to do is for you to calculate the missing SNP in your data. That's why you have plink.lmis. So it generated to the last command that you ran generated to output plink.lmis and plink.imis. All right. So the difference between plink.lmis is that plink.lmis, L for locus, basically contains the SNPs that are missing, the number of SNP missing. All right. So if you look at this, when we did plink.lmis add, you see the first column is chromosome. All right. The second column there is SNP ID, where you have RSID. Please, can you guys sit down, please? Thank you. All right. So you see, you have SNP where you have RS. If you look at the second row, where you have 1 RS 218. So that is a SNP ID specific to each SNP. All right. Then after that, you have NMIS. So the NMIS shows the number of missing SNP. All right. So you see, RS, the first SNP there is not missing. That's why you have 0. All right. So when you look at the one that has 4, that means 4 of this SNP is missing in the data. So that's why you have 4. So N means number of missing SNP. All right. So you have N geno. N geno is the number of non-obligatory missing SNP. So what that means is that sometimes your SNP could actually be missing not because of genotyping error. All right, it could actually be that it wasn't really called or imputed. All right, so that's why it is missing. So it is called number of non-obligatory missing SNP. So that's why you have N geno. Then the last one is F miss, which shows you the percentage or the proportion of the missing SNP in that particular data. So that's why the one that has 0, it's also 0 like that. So I believe we all understand. All right. Now the same thing is just do plink, just do add plink imis. So let's see the number of missing sample. Plink add, just do add space plink dot imis. Also what you're doing is you're doing add, all right. Then you're copying plink dot imis. Yeah. Okay, let me just run that. So can you see the result of the plink.imis? So the first column there is family ID. All right. Like we said, that you have 3 types of plink format file. You have the FAM file, the BIM file, and the BED file. All right. So most of the time, the family information is always created in the FAM file. All right. So you see, the first column is the family ID. The second column there is the individual ID. So that means when you do, for example, you want to do a GWAS of everybody in this class. All right. So what you just need to do that each person will have their individual ID such that if anything happened to such individual, you can easily trace it. And again, maybe you want to use it during diagnosis and all those things. Sometimes people might be having some ailments and they are not really aware of that. And maybe you are doing a diabetes study and then you discover that, oh, this particular individual may be having diabetes or something. And then maybe in your consent form, you say that you might disclose if such individual have that thing, you might disclose it to them. So you can easily, with the individual ID, you can easily trace such individual. And if you want to share the result of your findings with them, you may be able to do that. So that's why you have individual ID. Then you have missing phenotype. All right. So that's just telling that if a phenotype is missing in that data or not. So you have N or yes and all those things. And the next one is the number of missingness. But in this case, our number of missingness that we're talking about is the number of missing SNP in this particular is NMIS. So that means in this individual, you have 4,203 SNPs that this individual is missing. All right. So like that, so you could see the number of missing. Then the next one, like I said, NGNOS, the number of non-obligatory missing SNP. And then you have FMIS, which is the proportion, or you would say percentage of the missing SNPs in this particular individual. Okay. All right. So that is the first QC. Then we're going to move to the second one down. So are we on the same page? All right, a question came here now. We've been talking about SNP. Does anybody want to tell us what SNP is, by the way? Just raise up your hand, anyone, I'll just give you the mic. Okay, yeah.
The SNP is single nucleotide polymorphisms. That's changes in a particular unit of a nucleotide of a sequence. If you look at a sequence, if there are changes on a particular nucleotide, maybe you have GTT, GAA, and when you're aligning the sequence, you can look at which one has a modification or is change, or either deletion or whatever must have happened.
Right. Thank you. That's beautiful. So just to add to what he said, when you're talking about SNP, all right, what he had explained is single nucleotide variation, all right, such that when you have maybe a variation in a particular nucleotide in the DNA, but when such variation is you have like more than 1% of such variation in a population, it becomes a SNP. All right. So do you understand the difference between SNV and SNP? So basically, SNV is when you have mutation in a particular nucleotide. But when such mutation, you have the proportion of such mutation is greater than 1% in a particular population, it's no longer a SNP, but becomes a SNP. Right. So that's the difference between SNP and SNP. So that's why you're seeing RS. So when you maybe you can do that later. When you do RS21, you'll see the nucleotide and then you see the percentage of that particular, maybe the frequency of that particular SNP in different population. You'll see African population, Hispanic, European, and all those things. Yeah. Any other question? Yeah. So we'll move forward to the next one. All right, we're going to be moving to the next one. So what you've just done is you just have a picture of what is missing. You've not deleted those individuals. Okay. So the plink.lmiss and plink.imiss just gives you an idea of what is happening in the data. Now we are doing quality control. We don't need individuals or we don't need SNPs that are missing. So what we want to do is delete such SNPs. All right. So to do that, I'm going to be typing the command. So I'm clearing this so you can see what is happening. So I've cleared that. So the next command for us to do is we want to delete individuals that are having missingness of greater than 0.2. All right. 0.2. That is, they're having a missingness of like more than 20%. All right. So the command line you're running it's in there. So just copy it and don't forget, put ./ before plink and then you run it. So I'm going to run that now. So like I explained earlier, the first command you're seeing there is plink, which is practically you're calling the program you're using. All right. Then the --b file is a flag that specifies your data, the name of your data. Like in our own data, the name of this data is appmap3r3_1. All right. Now the next command line you're using is geno. All right. So this is a flag in plink that you use to delete missing SNP. All right. So what comes after geno is the number of SNPs. Is the SNPs having a proportion of 0.2. That's why you have 0.2. Sometimes you might decide to have 0.5 or 0.02. But the thing is, it's always good for you to start with a lenient threshold. So that's why we are doing 0.2, which is 20%. This is, you have to be very careful. For example, you don't have a lot of data, all right, and then you're doing 0.2, which is quite very too linear. So you might delete a lot of things. So we're going to start with this 0.2 just to delete SNPs, and then you see what happens afterwards. Then the next flag is make bed, which is a plink command that you use to generate, when you do make bed, you're still going to generate plink format files. So what you're saying is, when you look at this file, we're looking at SNPs with fnmiss of 0.2. Then when you get such SNPs, make files in plink format, that is bed, beam, and farm, and they named such file appmap3_r3.2. So when you run that, you see what happens. So you see it's running. All right, when I do ls minus lrt, what I am expecting to see, all right, is because my output is appmap3r32, so I'm expecting to see appmap3r32 with bed, beam, and farm. All right.
Can I talk? Okay. Hello, everybody. I hope you guys are following the class. Please let me have your attention for 1 minute. I'm sorry to distract you. So we just came from the VC's office. We introduced the team and I'd like to introduce our sponsors. Please, who is the sponsor for this conference? So, so please, can we put our hands together? Yes, so we have 2 people from 54G, and there's going to be 1 more person that will join us tomorrow. So one of them is going to address us briefly, just briefly. So let's introduce Mr. Julius Seramo. He's from 54G. He's the one with the face cap. So ask him for extra if you want one. The cap is fine, right? Okay. Then we also have Dr. Bukola Oyedele, also from 54G. Please stand in front of the camera. We have people on YouTube and we have people on Zoom that would like to say hello to you. She's pretty and she speaks Fone. So I'm trying to tap from that anointing. So me too, I can be posh. And I will not be doing like village people like you guys. Good afternoon. Good afternoon, everyone. It is my pleasure to be here on behalf of 54G. My name is Dr. Bukola Oyedele. As she mentioned, I research data insight lead. So basically, what I do is I take phenotypic data, which is all our epidemiologic data, public health data, social demographics, all those kind of stuff, and we provide it to your GWAS so that you guys can match it for what you guys need. So what you guys are learning today and throughout this conference is actually really, really important. While I think our work is more important because you can't do anything without our phenotypic data. Yes. We also can't do much beyond just saying what we think is happening, right? So our phenotypic data just correlation, it just gives us some associations, but it doesn't give us the causation. And what you guys are doing is able to then give us the causation. So thank you so much for attending the training. And please, of course, do everything you can to learn to be the best. And we pray for more opportunities for you to be able to also then use this for work in the future and then to provide solutions for Nigeria and for Africa at large. Thank you. Hello, YouTube and Zoom fans. Thank you.
Thank you. Sorry for the break in transmission, but it was needed, right? From all the programming commands. So enjoy. So we were still going to take a picture with the t-shirts outside, maybe after lunch. When are you going for lunch? When are you going for lunch? Lunch is not ready yet, too, so you can continue till like 2 o'clock. So tomorrow. Okay, so we agree to wear it tomorrow. All of us. No, but we wanted to take it for the training. You just put it over it and remove it. Like my own plan is just to wear it over this and remove it afterwards. It's not a big deal. I know, just try. You can wear it over it. Tomorrow we have a different plan. Thank you. Don't stress me now, you guys.
All right, that was a good icebreaker. So we can go back to the command and then. Okay, yeah, moving back to what we've been doing. So what we've been able to do is, like I said, the first command you ran was just for us to know the number of missing data and the number of missing SNPs we have in our data. You didn't do anything to that particular data, all right? So the next thing we now ran before we got here was for us to do run a flag, that geno file, minus geno 0.2. So why we're using 0.2 is just for us to delete SNPs that have missingness that is more than 20%. All right. And what I was saying is that 20% looks very linear and it's quite too, all right. So when you did 0.2, where is it? So, okay, let me just. So if you look at the command, you have B file, which is the APMA data file. They use the flag --geno 0.2. Then afterwards, they will added another flag called make bed. So the make bed flag is for us to have an output in Plink format. And like I said earlier, our Plink format, bed, beam, and farm. Then what you're expecting when you run this command is to have 3 files that are in Plink format. And in this file, we are expecting not to see SNPs with missingness of 0.2. All right, so when you do that, you see that then you do ls minus lrt. So your ls minus lrt is just for you to list what is in a particular folder in the order in which they were generated. Okay, when you do ls minus lrt, hello? So Any question before we move forward? Hello. So it's good we are now on the same page. So somebody raised an important issue there that it didn't really find any difference between these new files that we generated and the number of SNPs in the previous file, which is true. All right. If you look at the number of SNPs in the previous file and the number of SNPs in this file, you'll see that they are the same. All right. And that is because we initially decided to use 0. If you look at it, we use 0.2, 20%, which means that we do not have SNPs that are missing, that have more than 20% missingness in our data. So that is why you do not have any difference between the previous file and this previous file. But the next one we're going to be using, which is 0.02, which is 2%, you would notice that you have quite a number of SNPs that have more than 2% missingness. All right, so let's try that. So one difference between these is that, again, if you look at this command, it's quite very similar to the previous one that we ran. But the only difference is that is the flag that we used. All right. In this command, we're using --mind. All right. But in the previous command, we use --geno. And don't forget where we are coming from. We're coming from SNP QC and sample QC. So when you're doing SNP QC, what you do is you use --geno. All right. But when you're doing individual per individual QC, you're doing --mind. I don't know if you have a question with that. So what I had said was that the first command that you ran, which is blink --b file app map --geno, then you use 0.2, is similar to this. But the difference is that instead of using --geno, we are using --mind. So Geno deletes SNPs, while mind deletes individual. Okay, so we are trying to delete individual with missingness of 0.2. So when you do that, and then you're saying the output file will be appmap _3 r3 and then _3 again. So you see that. Are we there? Okay, so we're going to be moving to the next one. We've tried to use a flag 0.2. So let's try using 0.02 and see if anything changed in our data. Okay, so the first thing we're going to be doing is that to use Geno 0.02, which is different from the previous one, which is 0.2. So just how you've been running, just call your plink. So you can see the command line that I just pasted there. So I'm going to hit enter. So can anybody spot the difference if you have things missing? If you want to talk, you could just talk here. All right. If you're on the same page, you notice that when we use 0.02, look at the number of variants that were removed. We have 27,454 variants removed due to missing genotype data. All right, so if this were to be present and you did your associational analysis, it's most likely that you're going to be getting false results. And then if you remember the Manhattan plot that I shared earlier, where you had a lot of noises beyond the control. So these are some of the things that contribute to it. But by the time you remove these things, you're having a clean data that you're sure that your results are quite true. Okay. So the next thing we're going to do is we're going to run this command now. This was for SNP, missing SNP. Who can remember the command if you want to delete individuals instead of using Geno? Perfect. So just do the same thing. And where you have Geno, replace it with mind and enter, and let's see what changed. But however, you see that we are coming from the output from your previous command is always the input for your next command. So don't make that mistake. What we generated here now is AppMap 3R34. So that's gonna be your input for your next command, and your output just change 4 to 5. I'm not sure, but let me just run it. Am I clear? Yeah, you use mind instead of Geno now. --mind 0.02. Then you change then the input to be AppMap 4, then output to be 5. Yeah. All right, ladies and gentlemen, I think we are done with the first quality control, which is missingness. Yeah. Okay, so I think we are done with the first quality control step, which is missingness. So what we've been able to do in the past 40 minutes was to do quality control missingness for per sample missingness and per SNP missingness. And that is what we have done. So the next one we're going to be doing is sex discrepancy. All right, so I'm going to explain that before we start running it. So because you're working with perhaps a large population, hello, can we all hear me? Yeah, so because we are working with a large population, there is a tendency that when you're collecting sample, they might actually be mixed up. Maybe an individual ABC is a male, but maybe that person is a male, but when you are labeling the sample, you erroneously labeled out such individual as female. All right. And then you have what is called, when you do Plink, Plink also has a way of determining the sex of an individual. It uses what we call XHA, X chromosome heterozygosity estimates. All right. So what happens is that males have XY chromosome, right? So what you can say now at the end of the day is that every SNP on the X chromosome are homozygous for male. So therefore, every SNP on the X chromosome that are homozygous for male, when you do the XHA, that is the X chromosome homozygosity estimate, such individual will be labeled as 1. So when you run your Plink and it estimates the XHA, which is, it can be F, sometimes called it F, and it's 1, all right, such 1 is male, all right? So, but when it estimates 2 is female, all right? So sometimes it then use sometimes 0.8. So anything greater than 0.8 is male, but anything less than 0.2 is female. So that is how Plink uses XHE to determine the sex of an individual, all right? So what now happens is if the recorded sex, maybe when you're collecting your sample, is different from the estimated sex, then you will see your Plink saying, oh, there is a problem because there is no match. All right. For example, if your reported sex is 1, which is male, all right, and then your XHE estimate gives you 2, then you're going to have problem. It's going to write problem. You're going to see it now. It's going to write problem. So what you're trying to do is you're trying to make sure that your reported sex and your predicted sex are the same. So that's what we're going to be doing next. And I had explained, it could actually come when you're collecting your sample mislabeling, anything can actually happen, all right? So that's what we're going to be doing now. So I believe everybody have done the first QC. So we are going for the second one, which is sex discrepancy. So any question before we start running that? Good, no question. So we'll run that. So yeah, that's a good question. So he's asking that, okay, we said that if it's greater than 0.8, it is male, and it is less than 0.2, it is female. So what happens between 0.2 and 0.8? It's gonna say 0, like no sex for such individual, because it's either you're a male or you're a female, no middle ground. All right. So you may not, I don't think you can have such figure. Hermaphrodite. Well, that's a good question. I'll have to go look into it. Yeah. Sex for such individual. Yeah, it does. Because either you're a male or you're a female. Yeah, so there's no middle ground. All right. So, and like I said, most of these things don't, yeah, it's XHE, like I say, X chromosome homozygous. So everybody's either you're female or female. So it just compute that based on the 0.8 and 0.2. Yeah. Okay. So we're going to run that now. Everyone is quiet. Okay, if you look at the working sheet, the protocol, you'll see we have sex discrepancy. And there is a flag there that is called check sex. All right. So all you just need to do is just copy that and then you check your sex. If you run that correctly, you should generate a file called plink.sexcheck. Plink.sexcheck. Are we there? Yeah, if you do ls minus lrt, the last file that was generated, plink.sexcheck. So you can just do a head of that file, HED, head of that plink.sexcheck. Yeah, the last one is plink.log. Yeah, the log file is, it just tells you what you ran, like, okay. And you do cats, let me just show what is in there. Cats, CAT, space, plink.log. CAT space plink.log. Cats, cat for cats. Yeah, space plink.log. LOG, the last file you generated. Now, while we are viewing that plink.log, it asks that what is plink.log and all those things. So any command you run will always generate a log file. So the log file just tells you, it's like it contains the program, the command you ran, the file that were generated, what you use as your input file. So I just wanted to say what is in there. So for you to see what is in there, you can use cat, CAT space plink.log. LOG, the last file you generated, plink.log. So it's just a log file of what you ran. So you would notice that for every command that we've been running since, you've been generating a log file. All right. So, and it's always very good. Maybe you run an analysis and you didn't run correctly, and somebody is asking you, what did you run? Can I see what you run? So it's just easy for you to just send them that log file. So with the log file, the person can easily say, okay, this is what you ran. This is probably where the mistake is, and this is what you should have just done, and all those things. So you will see you're generating a log file for everything. All right. So that aside, so can you do a head of plink.sexcheck? HED space plink.sexcheck. Head space plink.check. So head is just like a Linux command for you to say the first 10 lines of your head, HEAD. So I'm just going to type that, yeah. So I'm going to do a head of the pink says. So like what we've been saying, if you look at that file critically, you'll see that the first column, you are having family ID. We've explained that you have individual ID and you have predicted sex, PED sex, which was the sex that was collected, right? When you did your sample collection, maybe when you filled a questionnaire and you're asking the person, are you a male or are you a female? And the person says, oh, male or female. So that PED sex is the collected sex, the one you collected initially, all right? So the next one is the SNP sex, which is the genetically predicted sex based on XHE, all right? So the next one is the status. Are we having a match between our predicted sex and our SNP sex? So if there is a match, you have okay, all right? And the last column is F, which is like I explained, the XHE estimates, all right? So what we are concerned with is that, are we having any individual in this data, okay, that is having problem with sex? So the best way to look at that is we can just quickly do a grep. Grep, like you know, what I said was that when you have predicted sex and your SNP sex, when they are not in accordance, you have your status is going to be a problem. But when they are in accordance, when there is consistency, it's going to be okay. So these ones that are showing using these 10 lines, we have all of them being okay. But when you do a grep, GREP, all right, GREP, you can see the bird, GREP, problem, all right. If you look at what's the command we just ran, we are looking for individual that has problem where your predicted sex is not the same as your SNP sex. When we check the head of plink.sex check initially, you see that the 10 line, everything seems to be okay. We had okay, 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 okay. So we want to look for individual where there is no consistency between predicted sex and SNP sex. So when we just do a grep problem in the last part, the plink check, it's just a very quick way. Instead of you have a lot of lines. So instead of you to just be looking at them, and especially if you have a lot of SNPs, you don't want to go through the test. So it's easy for you to just do a grep, then grep problem plink.sex check. So when we did a grep problem plink.sex check, you see this individual NA10854, they said this person is 2, female. But genetically, this person is a male. So there is problem in that place. So that's why you have problem. So in this case, what you will probably do is, you may, if you're really still interested in this individual, is to go back to the person who collected the data and confirm, okay, is this person a male or a female? Right. So you see why we are doing quality control, because you're giving us a picture of what is really happening in our data. We couldn't have just gone ahead and used this, which is going to bring a problem. So you see, when you did a problem, bring the sex check, you're having a problem. So any question? So what you must always watch out is that your predicted sex and your SNP sex should always be, your genetically predicted sex should always be the same.
In the first flag, there was no mismatch. There was no mismatch now. Everything was the status for every one of the.
Yeah, so why you're doing that is, like I said, okay, let's do WC. Can you do WC minus L space sex check? No, plink the sex check. just wait, just do this. And just do playing. That's the enter. So you have 166 individuals. So you don't want to be looking at them manually. So that's why you just quickly grabbed the one that has problem and then you found it there easily. Okay. This one that has problem, the family ID is this, why the individual ID is this. So that means you just grabbed this from this old file. So this is inside this particular file. So you just brought it out of that file. Yeah. So hello everyone. So that was what happened there. So the next thing we want to do is let's go to the next command. So if you run this command, grep problem, ping the sex check. Okay, let me just run that. Do it. So if you look at a command on the board, I decided to split that command into two so you could understand what when we run the command at once, you will have an idea of what is happening. So the first command that we ran initially was to do grep problem ping.sex check, which was also to have the SNP or individual having problem. So now we now combine that particular command with another command. So what we now did was you did grep problem ping.sex check, all right? Then we use, then you do awk, right? So what awk does is we're trying to print column 1 and column 2 from plink.sex check, all right? So if you look at plink.sex check, when we did the head of plink.sex check, our column 1 and column 2 is the family ID and the individual ID. So when we now run the next command, which is awk print column 1 and column 2, where you have $1 and $2. So it's going to print the first column and the second column of plink.sex check. Then when you say, after you've printed it, pass it into a new file called sex_discrepancy.txt. So when we passed it, and then we had when you did the head of sex_discrepancy.txt, you see that what we have there is column 1 and column 2 taken from plink.sex check. I don't know if you understand. So I'm going to take it again. Yeah. So let's look at it from where we had the first command, grep problem plink.sex check. So when we ran that command, we saw the individual that is having a problem where there is inconsistency, discrepancy in the sex. So we now decided to run the command at once. So the whole idea is we want to look at this individual and put it in a new, put it in a new file such that we can use that file next in our next analysis. So what we now did was to do grep problem from plink.sex check. There's nothing different. It's just what we ran initially. All right. So we're not saying from this plink.sex check, can you print column 1 and column 2? All right, from plink.sex check. So when you print it, pass it into a new file and name that file sex_discrepancy.txt. So that was what we had. So when we now generated that, that file, when you did a head of sex_discrepancy.txt, we eventually had the first and second column that were taken from the plink.sex check. Okay. Yeah, even if you remove problem, it will still run. Yeah. So we'll go to the next one now. Yeah. So what we're going to do next is that we want to remove this individual that is having sex problem from our file. All right. So what you need to do is so if you look at a command on the board, I have plink --b file. So our input file in this case is appmap3r3_5, right? Then what we're now doing is we are using an additional flag, which is remove. It's a flag in plink So what we are removing now is this individual with sex discrepancy that we have put inside this sex discrepancy.txt file. All right. Then you're not saying that once it is removed, make another bed file and call it AppMap 3R36. So when you hit enter, so if you see what we just did now, our input file, which was AppMap 3R35.farm, the total number of individual there is 165. But when we now run this command, remove sex discrepancy text, our result, which is, our output now, which is AppMap 3R36, now has 164 individuals. So you've successfully removed that individual that is having a sex discrepancy. Okay. Hello, everyone. So, okay. So what we've done is, if you look at the command that we ran initially, we generated sex discrepancy.txt. All right. So we now have that individual in a file. So the last command you ran before you generated this was to remove that individual from the overall file. So that's what you did /blink --b file appmap 3 r3.5 --remove. All right. So when you remove, then the next thing you put was sex discrepancy.txt. So what we're trying to do is we want to remove this individual from the overall file. So when you removed it, you generated a new file called appmap 3 r36.farm. Now, when you do a word count of the previous file and the new file you generated, you will notice that the previous file, which is appmap 3 r35.farm, now has 165. I mean, it has 165, which was the previous file. But when you remove that individual with sex discordancy, you now have 164. So that means you've successfully removed individuals or individual with sex issue. So you've done your second quality control, which is sex discrepancy. Okay, any question? Yes. Hello, can we? It has to be issue with. Okay, so hello, I think we'll go for lunch now. So we can continue the remaining QC step afterwards. Hello, everyone. I think we can go for lunch now. Then we'll see. All right. I think what we're going to do now is when we come back from lunch, we'll do the remaining QC. But in the next 20, 30 minutes, should in case you have a new problem, then we can just use this opportunity to fix your problem so you can be on the same page with everybody. So the next 20 minutes, anyone who have an issue, then we can fix that. Yeah.
Can we start, everybody? I hope you can hear me very clearly. Can somebody please confirm if you can hear me? Can everybody hear me? If you can hear me, can I see a wave so that I know you can hear me? Okay, people at the back, can you hear me? No.
We can hear you.
Okay, so thank you. So welcome back, everybody. We're going to start the second section and the last session now. So we have the instructor for that section. So this session will be focusing on the polygenetic risk score analysis. And we have Dr. Chikowore that will be taking that presentation. But before then, I would like to know if we have the team for 54 Gene in the class so that I can relay the message from the participants as well as the member of the Nigerian Bioinformatics and Genomics Network to them. And I would like this to be official and pass it across to the leadership of 54 Gene. So Dr. Oyedele and everybody for 54 Gene, welcome once again. And thank you for taking the initiative to support this workshop. We are so excited that we have so many people on this call. This morning we have 55 people in the class, 25 people joined by Zoom, and 10 people on live on YouTube. That was 90 altogether. That was very encouraging. But unfortunately, this workshop is only an introduction, only for one day. We are planning that in the future, we're going to be organizing 1 week long workshop, and we're going to be counting on 54 Gene to support that. So one question that is very common is people are looking for opportunity where they can do internship. People are to do analysis for genetic analysis, bioinformatics analysis. People are looking for opportunity for postdoc. People are looking for opportunity for PhD scholarship. And that question keep on coming. I would like you to just, in 1 minute, if you can speak to that and tell us what kind of opportunity are available in 54 Gene for people that are here today and other people that are not here, that are building their capacity to be able to analyze large-scale genomic data.
All right, great. Thank you, Dr. Fatima. Again, nice to be here. 54 Gene has, through its ACTG platform, scholarships for both postdocs and PhDs, or vice versa, PhDs and postdocs. So if you are here and you are pursuing a PhD, or you are done with your PhD and you are pursuing a postdoc, there are numerous opportunities that are available through our fellowships, through our grants, through our scholarships that are available to you now. It may not necessarily be available this year because we've already gone through the rounds for this year, but it will definitely be available next year. So please come back and check. And we're also, going to be creating more opportunities. The most important thing for 54 Gene is the idea that we are enabling people, we are increasing capacity in every way, shape, or form. So even if, for example, you are not seeing something that might work for you, send an email, contact us, and let us see what opportunities are possible, what can be created. This is definitely one of our missions to ensure that we, and by we, not just the work that we are doing, but the work that you are doing, are able to increase capacity to enable us get to the point of translational medicine where we want to get to. Thank you.
Great, thank you very much. So we're going to start now. In the morning, we have been doing GWAS, and with the participants have been working on doing the analysis on their computer. So this afternoon, we're going to be doing polygenetic risk score. So we have our colleague, Dr. Tinashe Chikowore from South Africa, that will be taking that particular presentation. So I'm going to put on my video now, and I will hand over to Dr. Tinashe to take over. So you can start sharing your screen. Please feel free to introduce yourself, Dr. Tinashe.
No, thank you so much, Professor Shegun Fatumo, and thank you so much for the sponsors of this prestigious Congress, and also for the organizers and also Landmark University for making this feasible. My name is Dr. Tinashe Chikone. I'm a Wellcome Trust Fellow, affiliated with the University of Witwatersrand in South Africa. I'm also a partner and collaborator to the Africa Computational Group that is led by Professor Shegun Fatumo. So yeah, I'm so happy to be taking you through an area that I'm passionate and interested about, which is with regards to polygenic risk scores. So I'm just going to be sharing my screen such that we can take time to learn together and interact. Okay, and feel free to ask questions or even write them in the chat such that we can go through them as we go along the way. So I'll just start with this. I mean, I think some of us might be fortunate to know the stock market or the stock exchange where people will be selling shares and transactions will be happening. We also envisage one day that even in a clinical setting, things such as polygenic scores will be run, you know, when participants get inside. We might have a well-structured clinical setting that can actually take out information from the genetic side and other various omics technologies and enhance how we render treatment. So before we go into that futuristic perspective, it'd just be key to say, where did we all begin with the research of polygenic risk scores? So I'm going to take you through the background of polygenic risk scores. We're also going to learn some key concepts using a single approach. This is an introduction, so we'll start with this approach about the pruning and thresholding approach, and all those key aspects you're going to see some demonstration through the presentation, and you're also going to actually run them through the practical. Then we'll also give you examples of other polygenic risk score approaches. So where did this all start? So around about 2004 to 2009, with a wave of genome-wide association studies. Then there's a guy called Sean Purcell, the one who actually developed Blink. He just said this idea to say, wow, what about if we just combine these variants into a simple score? So he wrote it in the paper that's on the other side there, which is the common polygenic variation contributes to schizophrenia and bipolar disorder. So that work was published then. And many works later followed up, even us with other colleagues. We also wrote something in the African side. And, you know, the field evolved. Initially, people were looking at signals which are only GWAS significant. Even when we published, we were also playing around those terms, and they were called genetic risk scores. But however, around about 2014, this perspective started to change, that we need to go beyond the limit of variant detection and use all the SNPs that you might find when you do a GWAS. So we later on started seeing scores which have 2 million SNPs and more. But why are people so fascinated about this polygenic risk score? So let's go back again to the GWAS that we were learning in the morning. So for example, a person might have a variant called T, and this T is concentrated in the cases and less frequent seen in the controls. And when you do your GWAS through your Manhattan plot, you pick that signal. But in reality, when you do GWAS, we're not looking only at one variant. We actually have millions of variants. With the imputation that you have learned in the morning, you will find your studies that have 13 million, 20 million, or 30 million variants. But however, each variant only explains a small effect size, meaning when you run your simple linear regression, you can find the effect sizes are so small. But however, when you combine them into a score, they also make us see another perspective. So all these scores, they actually follow a normal distribution. And this normal distribution enables us to stratify people, to say these are people who have a higher genetic burden or who are highly at risk, looking definitely on a polygenic score. And when we look at that, then we are able to also see that there are people who actually also have a lower genetic susceptibility, those that are indicated in the green there. We can even look at these things through percentiles. So what we'll be doing with percentiles is, because it follows a normal distribution, it means we can actually split all these data sets into 10 equal groups. So this will be same numbers to say people who are in this group 1 up to 10. And this enables us to stratify people again from low to high risk. But you might be asking, what would this help us? Actually, people are actually saying that it can even help, even when a clinician is want to make a decision. So for instance, this was a clinical trial where they stratified people based on a polygenic risk score, those in high risk and those also in low risk. So, but those people were the high risk according to the polygenic risk score. They also found that other things like they had a threefold higher. Some of them had actually experienced threefold higher microvascular events, like 2-4 higher worsening nephropathy and the 4-fold higher cardiovascular possibility of actually succumbing to CVD when they're comparing them with those in the low risk. So when they then did this intervention, they then realized that this intervention was only beneficial to the people who are high risk, people who are about to highest risk of CVD or even complicating, but it was not even beneficial in this to those people who are low risk. So we can see here that polygenic risk scores can actually help clinicians to make up decisions on who should receive a particular treatment, something that is quite key and something that we are looking forward to see it happen even in the African context. But before we get so pumped up, we also want to look at what has even made this very, very much fascinating. So if you can look the news readers, I mean, there's a study that was published about polygenic risk scores, and many people were writing, and they say new tests predict the risk of serious disease months before symptoms appear. So this is also the advantage of genetics. Remember, with all these classical risk factors, you need to see someone becoming obese. You need to see someone manifesting, maybe having a high blood sugar and all that. But with genetics, I mean, people get these things when they're born. You can even trace way before they actually manifest with any risk factors. Then the other thing that's also amazing, when people were really fascinated about a study that had been done in Harvard there. So as the news reporters are writing, so someone said the Harvard scientist thinks he's a gene test for heart attack risk. He wants to give it away for free. So it's true. You'd find that most of these polygenic risk scores, there's what we call the PGS catalog, where what people have published, like what you might have learned in the morning, that when you do your GWAS, you actually ask to submit your summary statistics to the GWAS catalog. There's also a polygenic risk score catalog. So these guys had done a seminal work where they actually showed that using just that score, you could actually even identify people, just as you identify people with monogenic disorders. And this was tweeted a lot. It was shared a lot. News writers wrote a lot about it. And I mean, everyone was just so happy about genetics. GWAS again to say clues to your health are hidden in 6.6 million spots in your DNA. So this was a score that polygenic risk score comprising of 6.6 million variants. But however, let's really look at this closely. So this is in the European setting. But when you come into the African setting, it's something else. So they actually say that when they compared prediction of a polygenic risk score trained in a European setting and tried to apply it in an African setting, it had a 4.5-fold less prediction. Why is it so? It is just because that the majority of the GWAS are actually coming from individuals of European ancestry, and only about 1.1% of those individuals are Africans. But if you look here on the scale here, in terms of global population, you can actually see that the Europeans actually occupy a smaller percentage, I mean, of the global population. But however, actually the majority in the 90% has only been done in Europeans. So this also is a challenge, but it's also something which is an opportunity. Why is it an opportunity? It means that, I mean, people who are working with African data have that ability also to generate, I mean, polygenic risk scores without having to compete with people from the European side. But also it's a challenge then that if we don't generate numbers, because the advantage here is on the European or on the Western side, Professor Shegu's paper, they only had 14,000 participants. So meaning we yet, I mean, to upscale our work to ensure that we get there. But however, there are interventions and programs and initiatives that are in the continent, which hopefully will make us reach there. Right, so with all these things, we have seen the poor transferability, but also another amazing thing is there's great genetic diversity in Africa. And also this great genetic diversity, you can actually see a PRS does not perform the same. So this is a work that we were doing here. And we actually saw that when you are predicting for BMI, for instance, it predicts better in the South, not in the East and not in the West. So this is also as a result of environmental and genetic differences. So there is also a challenge here. A challenge means that one polygenic risk score will not work in the whole of Africa. But maybe it's just a statistical issue that can be solved. So we see there are really new opportunities, I mean, new consortia that are working to optimize polygenic risk scores in underrepresented populations like Africa. So who knows? I mean, as you are sitting here, maybe a PhD student or a master's student, this might actually be a potential topic that you want to explore to say, fine, how can I optimize, come up with a score that can maybe work for the whole of Africa? Something interesting, because remember in Africa, we have limited resources. So we might not have that luxury of saying every country has its own polygenic risk score, but we might just get something that works for everyone, something which is a food for thought. Right, so with all that, let's then get into the real nuts and bolts of what we want to learn today. How do you really come up with a polygenic risk score? So what do you need? You need what we call a base data set or a discovery data set. So this is something that is larger, and you also need an independent data set. That is where you want to train your scores in. So polygenic risk score is just a summation of you are taking your SNPs, then you are taking the beta. So this is your effect sizes. So for instance, when you do, so the simplest thing about genetics is just to remember your linear regression or your linear models. Then you will never get lost. So here we are saying the beta is coming from the discovery data set, which is the base. Then the SNPs are actually coming from the target data set. So then what we're just initially doing, so if you have a continuous trait, definitely your betas are easy to get. If you have a binary, it means your effect size is actually in the odds ratio format. But however, most softwares are able to convert to the beta scale just by getting the log of the odds ratio. Then your allele, your SNPs, you have alleles like genotypes, you just code them to 0, 1, and 2. So in essence, what you're doing is you're multiplying the scores, the SNPs, the alleles by the betas, such that you then sum it all. And each individual just has a single value, which is a score. And we have learned previously how we do that we need to do our QC. So the QC is just the same as you have learned before. These are just various ways of doing it. So we can do an LD adjustment, which is what we'll focus for, and also p-value thresholding, whereby we are going to select, because remember, previously people just used to think that the genome-wide significant is the best, but now it's an open space. We don't know which is the best p-value. So we have software that can actually compute polygenic risk scores at various p-values, then it will select the optimum one. Then in the end, we can then use our PRS now for association and also for evaluating. But it's also important to validate it in an independent data set. So there are actually nice tutorial guides, of how you can even go about this. A lot there's tons of this information in the literature, but actually the gold standard is always to make sure that your target data set and the best data set are different. In essence, you can't run a GWAS in one data set and also use that same data set to compute polygenic risk scores. You need this to be independent. So meaning the betas should actually come from a separate data set from the ones where you are getting your SNPs that you'll be using for computing your polygenic risk score. There can also be issues of strand misspecification or ambiguous SNPs, and softwares also work with this. And there are many other methods besides the one that we're going to show you today, which is called PRECISE. We also have other machine learning approaches that have been evaluated. So in essence, you'll be just using a simple workflow. So for instance, you need to make sure your best. So your best, this is your discovered data set. Has it been QC'd? So you check to make sure are the alleles well indicated well. You also check, does your base of a particular single trait or is it a multi-trait? You also learn this in advanced approaches. You need also to QC your target data set. You have been learning how to perform QC in Blink. Also, these things can actually be done, like removing sex mix matches. You've actually looked into it. Then you also ask yourself, do you have multiple best GWAS in different ancestries? Remember what we said, poor transferability. So if you have none, you can just run, the simple ones. If you have, then you need also other optimum approaches like a multi-PRS or other PRSs that have been formed, because we know the majority of the GWAS is from Europeans, so it will tend not to perform well. But nonetheless, people are just using what we have. We cannot wait. While we're waiting for African data, we're also using even multi-ethnic. So you can still run these approaches like PRECISE, LDPRED, but today we'll just show you a PRECISE how you can use it. Right, so let me just get straight into an example here before we break for the tutorial. Right, so let me show you how your results will look like. So when you've finished running your using the PRECISE approach, you get a log file, you get a .precise file. We're going to look at it. You get a demo summary, you get a .summary file, and you also get a plot file. So let's first look at the log file. How does it look like? So your log file is in essence just showing us the commands that you ran. So it first tells you this software is called PRECISE. This is the commands that you ran in this upper part here. So you don't need necessarily to write all this code. You're going to see in the practicals that you need to specify which is your base, which is your target. So you can see there's binary target, the base, that is your discovery. So meaning you need summary statistics, which are going to be your base. You need your binary target, which is going to be your genotype data in Plink format. Then you can specify that I want to clamp it's 250, so this is just a window. Then you also are going to specify the LD correlation. You need also to have a covariate file because you're going to adjust for age, sex, and population structure. Don't worry about these intervals, it will generate them out. You will write the name. This is the name of my output file. The phenotype can actually be in a separate, you're running BMI, you're running blood pressure. Then based on what is in your summary statistics, it's going to write. So it's going to read the file. If there's a beta, then it says the stat is beta. Your target data set, that's the name of your genotype data. Then thread just means you own how many cores you want to run your analysis. So after it has done that, it will start reading your genotype data where it's saying starting processing. It's actually showing it's going to read this genotype data. Then it reads your summary statistics. So it says there are these million of SNPs in this. So you can see this is 24 million SNPs or variants that are in the summary statistics. Remember what we said, that ambiguous variants, it actually removes. So this is just ambiguous because these are the alleles that are difficult sometimes due to strand effects to make sure, is it really a C, is it a G? So the software just automatically handles them out such that your polygenic risk score, even when you share it to someone else, I mean, it can really work well. Then it will then say, okay, so after doing its QC, there are 20 million SNPs. Then it reads your genotype data. Sorry. So after reading your genotype data, it says, no, you have this number of people. This number of people have these SNPs. So we're going to include 18 million. Then it starts to processing. So at the end of it, it does some clamping. So what is clamping there? It wants to reduce the number of SNPs that are going to be used for the polygenic risk scores. So it clamps using LD, because remember, these SNPs are related to each other. So by just playing around with the relation, you can remain with fewer SNPs that can actually cater for the ones that you don't have. Then it runs a simple linear regression where it has age, it has sex, then that is what you're adjusting for. Then at the end of the day, it will show you your results. So which results will you see? So you see the dot summary file. This is just going to tell you in summary what you are getting from this analysis. Okay, let me just try to make this a bit bigger so that you guys can see it. Okay, so what this dot summary file will tell you that you are running this phenotype. Usually we don't specify, so this is the best. So the p-value threshold is just telling you your best p-value was the one which was less than 5 times 10 to the power minus 8. So then, because it has run a linear regression model, so the full model is which one, which has the PRS, it has age, sex, and PCs. The now model does not have the PRS. So for you to get the PRS, the variance of the PRS is just subtracting the variance of the model, which doesn't have the PRS, from the full model. Then you get the variance of the PRS, which is on this side. Then it even gives you the coefficient. I mean, it gives you the p-value of your PRS in the model. Then it tells you the number of SNPs that have been used to compute that PRS. So this is quite good because you would want to transfer it to give someone else to use it somewhere else. So you know, okay, my PRS only has 38 SNPs. And you're going to do in the tutorial how to even filter out those SNPs and even share it to someone else. Then we also have a file. Okay, so these ones will refuse to open. So we also have the bar plot here. So the bar plot will just show you the PRSs that you actually have generated, which one has worked well, which one explains the majority of the variance. So the one which has the biggest bar is usually the one which is explaining the majority of the variance. So we'll just jump to the terminal such that we can actually get to see these other data sets. Okay, just wait there. Right. Okay, I'll just maximize my screen here. So I think maybe today you have learned the issue of 5 formats. So there are certain formats that if you click, you cannot open. But when you are within a terminal, you can actually open them. Right, so let's look at these files. Okay, let me try to make my screen bigger here. Right, I think you're now seeing it. So I've shown you the log file. I've shown you the bar plot. I've not shown you the dot best. So the dot best file, if you do a demo there, dot best, this is how it looks like. So remember, the simplest thing to learn this thing is just to go and understand linear regression only. So you would find that what was in the regression, so these are the individual regression. So this is the score. Each individual has only one value. And that's what you're seeing there. That's the PRS. So meaning each individual actually is in value, which is part of the score. Then I'm also going to show you the dot precise. What is it? So in the dot precise, remember we are computing these p-values at many thresholds because we don't know which p-value threshold will give us the best score or the most predictive. So the dot precise has many PRSs. So meaning this application actually, for in this case, let me just check how many polygenic rescores were created. So it created 6,003 polygenic rescores. Then it ranks these ones to say which one is the highest p-value, the highest variance. So predictivity is measured in terms of variance. A good predictive PRIS explains more variance about the trait. So the one which is the highest variance is the one that is indicated in the dot summary. So these are just some of the facts that you get when you run precise. So when you are now running your analysis, you are just using this, the dot best. Then you can actually, it's now an additional variable. So it's now the PRSA, each individual has a value. So that's now an additional. So you can now use it in your future work, trying to see how this PRS works in your downstream analysis. So in a nutshell, yeah, this is what I can just, as an introduction, for you to see how these polygenic rescores are created. So I'll just open up for questions. Sorry, it's getting a bit dark here. Apologies. So I'll just open up for a few questions as we reflect on this.
Do we have any question? Please feel free.
Okay, it's a bit dark.
Is somebody speaking?
Sorry, any question? Anyone with question?
There's somebody at the back there.
My name is Omena Emmanuel from Ibadan. I want to request from the last speaker that we would like the breakdown of the lecture materials he has taught us this evening, and especially the use of the GRABLD machine learning to approach. And how do we minimize these polygenic risk scores? He said it has a poor transferability. What are its implications? Thank you.
Okay, no, thank you so much for the question. So yeah, so we actually doing current research, which actually has shown that we can improve the transferability of polygenic risk scores. So in work that we are doing, we've actually seen that if you use summary statistic like a discovery data set that is coming from a multi-ancestry study, that enhances prediction, especially in Africa. If you also use like summary statistics from African Americans, that also enhances prediction in also continental Africa. So there's work that's currently going there. So it doesn't mean you should then run away from polygenic risk scores because the European GWAS don't perform. There are also those two data sets that can work for you. So in terms of the machine learning one, the GRAB-LD, so there are many tools that have been developed. So it's also available. If you Google it, you find it. They actually have a repository. We can even share links for people who might just be interested to utilize it. And as indicated, definitely the presentations will be made available for people to look through them. Thank you.
So just to add to that, I think the person asked a question also wants to know about the implication of PRS. I think just to put in a very simple format or in a layman format is that if we are able to get PRS, that we can get PRS trained from one data, can we use that PRS to predict people that may have a particular disease in 10 or 20 years? For example, supposing that we have the capacity to be able to predict that, or this person, and this person, we have cancer in 5 or 10 years. That would be a breakthrough, because what will happen is that the government can be able to cater for those people, and be able to support them so that they don't develop cancer in 5 or 10 years.
Yes, and even insurance companies, I mean, in countries which run also medical aids in addition to that. For instance, you could find in this COVID pandemic, they were saying, do this, because it limits their cost, even companies. So it's not only the government, they can even use that information to even advise their workers, you know. Yeah, and there are polygenic risk scores like in many areas, like even cognition. Yeah, so really, as you say, it's going to be a game changer, and it's definitely going to be a game changer, actually. Yeah. People are saying this is prime time now for polygenic risk scores.
And this is the time for anybody that wants to do GWAS PRS. This is the time to join.
Yeah.
So this is prime time that things are. Yeah. You can much more get grants if you have the skills to be able to do PRS and GWAS. So this is the time. This is the flourishing time for genomics in Africa. I tell you the truth.
Yeah, indeed. Yeah. And the good thing is also, I mean, a lot of this genetics is still like African genetics is African genetics. That's the advantage, you know. So no matter how much is done in Europe, we still need to do more here. So this just puts us on a good pedestal to say, I mean, learning these skills, making the most of it, your career would definitely change. You won't like feel like, you know, there are certain things which, like, even if you learn here, when you go on the other side, you feel like, oh, I need to learn more. But if you establish yourself in African genetics, that's it. No, you grow there. Yeah.
Okay. Any other question?
Okay. I think people are tired, so maybe we can transition.
There's a question at the back. Yeah.
Thank you. My name is Hakim Sikiru from FUT Mina. My question is that when designing a study to identify some target genes as candidate genes for a particular physiological dysfunction, let's take, for example, immune dysfunction. We are trying to identify some genes as a target gene. Now, we want to use PRS to correlate with whatever regulation of those genes, whether if immune is compromised, those genes are upregulated. If immune is not compromised, those genes are downregulated, or things like that. So is the PRS going to come before the gene expression studies or after the gene expression studies? Thank you.
Okay. Yeah, so in your case, that's more like you want to understand function. So the challenge then is, will you find a similar study which is trying to do what you're doing? So definitely you wouldn't use polygenic risk scores there. So, because remember, you already have some hypothesis that these genes might work like this. You'd rather use, for instance, haplotypes or what have you, or even some gene-based tests whereby you group these genes together, or even do like pathway analysis to actually see how these genes together affect expression. So you'd really need, that's more like a functional study, and you wouldn't apply a polygenic risk score on that. Yeah. Because remember, polygenic risk score is to stratify people. It's not to understand expression, but you want to put people into groups based on risk. So each person is a particular value, a single value, then it helps you to put them in a particular group, whether they have a higher genetic susceptibility or they have a lower genetic susceptibility.
Great. Any other question? Okay, so if there are no other questions, I want to say thank you very much, Dr. Chikuwore, for that great presentation. We're going to hand over now to Okwe and the team to continue. I don't know how much of time you have left to do the practical session. It's not compulsory for people to finish their practical session today. You can get the SOP and practice during their free time. So the conference is tomorrow and Wednesday. If you have questions, please feel free to ask. The conference will start by, I will want everybody on site to be there latest by 10 o'clock and get ready. And the other people also from online, they're going to be joining us after maybe 10:30 or 11 o'clock. But it's good that we have everybody ready by 10 o'clock at the center. So for me, it's goodbye today, and I want over to Okpe to go ahead.
Thank you, Prof. And thank you, Dr. Tanashi. Okay, so we'll be going back to our tutorial. So in this session, Dr. Tanashi just taught us polygenic risk score and all those things. So now we're going to be doing the hands-on session of polygenic risk score. So initially in the morning we did our quality. So in the morning we did quality control. So ideally there are more steps to quality control that we ran in the morning. So there are like 8 steps to quality control. So we only did like 2 steps. All right, but however, we've shared the protocol so you can actually run the protocol because we can't go through all the steps now. So it's now probably spend the whole day here. So we're going to be putting a hold on the quality control, and then we'll be doing the polygenic risk score hands-on tutorial, and then we'll call it a day. Okay. So Brenda is going to be sending the file. So the file is called PRS file, just how you had GWAS file. So in this file, in this folder, you have all the files that you need. So we're going to be using Precise2, like you just explained. So in that folder that you're going to be sent to you, so you have Precise2 inside that folder, just how you had Plink in your GWAS. So we also have all the data sets. So once you get it, I'm going to run through the data and all the files inside the folder. And then one thing again in the folder is we have precise2.mac, and then there's another one, precise2 for Linux. So if you're using Linux, so you're going to be calling precise2 Linux. Don't use the Mac. So there are 2 files, precise2 Mac and precise2 Linux. So for people using this, just use Linux. And people who are using Mac, you use Precise2 Mac. All right. So once you send the folder, so we'll take it up from there. Okay. How many of us have the files yet? The photo. Okay. Good. So how many of us have the folder yet? Please can you just raise up your hand. One person, okay, 2, 3, 4, 5. If you have 10 people, we can start. All right. Hello, everyone. So just to save our time, it's already after 5. So I am sharing my screen. The data Brenda is going to be sharing is going to be having all these documents inside. So you are having a file called AFR, then you have AFR.beam, AFR.bed, and AFR.farm. Those are the plain formats for this particular data set. So inside this folder, you have afr.cov, which is your covariate file for this particular data set. Then you have afr.its. All right. So you have afr.its, which is your phenotype file. Then you have precise underscore mark, which is the for those who are having Mac. So instead of you running PRS underscore Linux, you're going to be using PRS underscore Mac. All right. Then you have another data its gwas.text, your base data sets, and I'm explaining for you to get the concept more what you mean by your base data set and your target data set. And then you have precise underscore Linux and then R script for precise. So we're going to be starting this now. Again, in the folder is this protocol, and then we're going to be using this protocol to run a demo for the analysis. Hello. For those of us that have the file, the folder, you have to CD. You have to CD into that folder, and the folder is called PRS open your terminal, and then do CD into PRS. You can move the folder back into your desktop. When you download it, it's probably going to be in your download. So if you don't want to navigate into your download, just move it back to your desktop. So just drag the folder and drop it on your desktop, all right? So once you drop it on your desktop, open your terminal and do CD, then PRS. So you should get everything, then we'll pick it up from there. What CD does is that it helps you move between folders, yeah. So when you do CD, so you're moving into that particular folder. So please, if you've not downloaded it, I would suggest you can share with somebody or just pair with somebody who has downloaded it and then share your screen together and then so that we can move forward. So let's run the first command. Like what has been explained, that you have your target database, your target data set, and you also have your base data set. So in this case, your base data set is the height GWAS.text, which most of the time comes in the form of summary statistics, while your base data set, while your target database, always comes as individual level data sets. So let's quickly look at how the base data set looks like. So just do add height GWAS.text. Okay, if you look at what is on the board, this is our base data set, and this is how most summary statistics data always look like. You have different columns. The first column is the chromosome the chromosome number. Then the second one is the base pair position of that particular chromosome. And the third column there is the SNP ID for each of the SNP. Yeah, I'm viewing the height GWAS.text. So just do add GWAS.txt. So if you look at it, so you have SNP, then you have A1. Your A1 is your minor allele. What minor allele? Yeah, minor. Then your A2 is your major allele. And you have N, which is the number of individual in that particular sample. So that is your N. That's the total number of sample. Then you have S, which is the standard error. After this was gotten after you've done association analysis. Then you have P, the P value, which shows the significance of each of the SNP in that data set. Then you have OR, which is the odd ratio. So the odd ratio is the beta effects of that particular SNP, and this odd you're having odd ratio here because this is a case control study. All right, so you probably run a logistic regression. So if it was a continuous traits, you would have beta instead of odd ratio. All right, so but when you have a binary traits, it's always recorded. The effect size is always in odd ratio, which is OR. All right, then you have IN info, which is the imputation score of that particular SNP. So that's what your data set looks like. And this is almost GWAS summary statistics. Always, I don't know if anyone has a question, then we'll move to the next one. No question. Yeah, so the M there is the Morgan, like genomic distance, like what we explained earlier, the genomic distance that explain the genetic ancestry diversity of that particular SNP amongst population or different species. All right. Oh, sorry, it's not M, it's MAF. So the MAF there is minor allele frequency. So minor allele frequency is, you have one there, which is the minor allele. So it describes the frequency of A1 in a particular population. So that's why you have MAF, minor allele frequency. All right, sorry, it's not M. I thought it was M. So let's run the next command. It's just a preliminary analysis to see the quality control flags that you can actually use in Precise. So I'm just going to copy this. So you're going to start using R scripts. So there's an R script that is also provided. So the first command you put in there is R scripts. Then after that, you put a path to where your precise.r is installed. So in this case, it's going to be on your precise file. So for you to get the path where it is, just do pwd. So the folder, the folder where you're working from, just do pwd, the present working directory. So it's going to give you the path to where your precise is. So just copy it and put it somewhere because you're going to be using it in the next command line that we're running. So when you type R scripts, the parts that you got, just copy and then paste it. Then you do /precise.r. So what you're trying to do, you're just trying to specify where precise.r is located on your system. So that's why you did PWD. So copy the PWD, copy the path and then do R scripts, the paths, then do /precise.r. Hello, everyone. So if you look at the flag that was run, the command that we ran, I think most people have gotten that to this side. So I can just quickly explain it. So I said we have a base data set. If you say we use the flag -- base ice.gwas.text. So that's our base data set. Then we used another flag -- targets. So our targets data set is always in individual level data sets, not summary statistics as our base data. So our target is AFR. Then we specified our phenotype, which is -- AFR dot height, then our covariate file AFR.COV, then our output is TRA trial 1. All right. So when you run that, so you see what we have. So it specified everything that we used. In our summary statistics, A1 is our small letter A1 and capital letter A1, minor allele, major allele, then the bar level, what our base data set is, what our target data set is. Our data is binary. Is it binary? If it was not binary, it would have written false, but it's binary. So that's why you had odd ratio. Then our BP, which is base pair position, is specified as BP. Then clumping threshold is 250 kilobyte. So what it's saying is that you're trying to clump within 250 kilobyte window. All right, so you're doing up and downstream 250 kilobyte for each of the SNPs. So that is the clumping, and then the p-value threshold is there. Then you've seen correlation coefficient, which is 0.1, and then the interval between them lower down. So once you run that, it's going to generate some files. So I'm going to scroll up, and then you see that file. All right. So hello everyone. From the look of things, everyone looks so very tired. Yeah. Yeah. Say yes. So I think what we've done, we've just done one of it. So if anyone wants to run it further, so just go through the protocol that we sent. And then when you run it and you have error or issue with it, we are still around, and then we can help you troubleshoot and see what happens. So yeah. So I'm going to be handing over to Professor Fatumo. So he has some words for us. So after the words, then we can call it a day. Professor.
You can stop sharing. Okay, so thank you everybody for attending to this workshop. So I know that we don't have enough time to finish everything that we want to do, but we've covered as much as we can cover today. So thank you very much for being here today and for attending. If there are any questions, please feel free to ask those questions. Feel free to send an email to us. We are all going to be available today, tomorrow, and also on Wednesday.
Sorry, Prof.
Yeah.
They can't really hear you, so he wants to increase the volume.
Okay.
Yeah. Okay, sir.
Okay, so thank you very much, everybody, for coming today, for spending your time with us and attending the workshop. We really appreciate your time. So I personally believe that you've learned one thing or the other. We're not expecting that you are already an expert in doing GWAS and PRS. What we have done today is just to whet your appetite. We are, like I said in the morning, we are planning to have a very much detailed GWAS and PRS course that would last for a week, that will provide the kind of skills that you are looking for. So I want to thank you again for coming. We're going to end now. So if you have questions, please feel free to ask your question. You can send an email. I understand that you have been given the protocol to walk through. If there are questions coming up, please ask them. So that will be the end for today. So tomorrow, as all of you have registered for the conference, because you are on site, we have already sent an email to all of you. Please try to be at the hall, not the way you have today, the main hall for the conference at 10 o'clock. We are expecting a few government officials. We have some distinguished professors that are coming. So tomorrow also we feature award for some distinguished Nigeria, 4 of them, including the Director General of the NCDC in Abuja, Professor Api from University of University, Dr. Abasi Eniobon, Forging CEO, and Professor Bewaji from University of London. So these people are going to be recognized and be awarded tomorrow, also as part of the opening ceremony. We'd like everyone to be seated at 10 o'clock. For everybody that will be joining us online, so we will ask them to join at 11 o'clock. So all this information are already in the email that we sent today. If there's any question, please let us know. Again, I know the LOC have already addressed everybody. There were some lapses yesterday in the where we are staying. I believe that will be sorted out today. So again, we apologize for what happened yesterday. So thank you very much, everybody. So we'll see you tomorrow. Let's be there tomorrow by 10 o'clock. Thank you. Bye-bye. God bless.